thank you very much, first and foremost, for taking the time out on this World Malaria Day to be part of this symposia. I'm really very grateful to all of you for taking the time out. I know these are very difficult times for everybody around the world, especially for all of us in India. And I really wish that all of you are safe and your family and friends are safe as well. We have a very tight program ahead of us, so I'll just get started by giving you very brief glimpses of what we've been up to in NIMR on and off due to COVID over the past year or so. And then we have a series of very spectacular talks by very, very highly eminent scientists and public health officials. And it's going to be a treat this afternoon. So I'm very much looking forward to it. And I really hope that all of you really enjoy this session as well. So I'm going to start by sharing my slides, which I hope are visible.
Yeah.
So as I mentioned, this is an International Malaria Symposia on World Malaria Day, which isn't today, but given the logistics, we thought we'll hold it today. And I'm just going to give you a few glimpses of the research that we've been initiating at NIMR and some of the strategies that I think are going to be important even for the discussion today that we will have. So as most of you know, and thanks to the national program, Dr. Avdesh Kumar, their most trusted lieutenant, is here. I'm really grateful to him for joining this. This is just showing the deaths from malaria over the past 20 odd years, and you can see there is a very, very significant decline in India, specifically since 2010 onwards, for example. And this has been really lauded all over the world by WHO, by India itself, and it has given a huge impetus and fillip to this idea that malaria can be eliminated from India. And I would like to again extend my great congratulations to the program for this really incredible job that they've done. And the research component of malaria, which is here to assist the program in its goal for malaria elimination, therefore has an existential crisis in a way. You know, where do we go from here if the incidence of malaria is really declining to this extent? And so the research themes that are important going forward have to be debated very well, because, you know, if it's a short-term research theme, that's understandable, but if it's in the long term, a decade or 2 decades, then there is probably not enough time to be able to contribute towards malaria elimination. The second key factor is that we are hoping very much that LLINs and ACTs, which have been 2 of the 3 drivers really that have reduced malaria all over the world, that they continue to work and don't fail. And I'm not going to really allude to the ongoing pandemic of COVID, but you know very well that one of the tools begins to fail for whatever reason, then the public health problem resurfaces. In this case, malaria can resurface. And we have to be very, very acutely aware that the going is as good as long as these 2 of the 3 primary tools don't fail. And so keeping this in mind, and also keeping in mind this sort of structure that NIMR had before I joined, we've initiated 2 very broad themes of which incorporate a huge amount of different sets of ideas between them. So we've gone back to the basics. We've broken up the problem of malaria in terms of looking at parasite, host biology, vector biology, and epidemiology on the whole, and it's called PAVE. And the idea is to pave the way to eliminate, but also to integrate. And here is the catch here, that, you know, the effort towards malaria elimination in terms of research that feeds into elimination work is equally important, of course, as is the basic research, because we need to be future ready. Again, not alluding to COVID, but as you know very well, if you're not future ready, even if you miss the boat by a few months and a few, in this case, mutants take over, you know how the system can collapse very rapidly. It is a very integral component to keep fostering the research that is targeted towards elimination. And we've made very nice thematic sort of windows around both MERA and IMM. I'm going to show you very briefly. Here is the MERA-India, the Malaria Elimination Research Alliance. We've had 2 rounds of, well, 1 full round of proposals that have been funded, and the second round is ongoing. And it's really encouraging to see here how widespread the proposals that were reviewed and funded have been from across India. And this is really heartening to see. There have been multiple partners in many of these research themes. People have come together with ideas that are central to their idea about malaria elimination, as well as consistent with the idea that NIMR has, for example. And so therefore, this is very encouraging to see in terms of all the thematic issues that have been rolling forward. And these are specific 2-year studies at most, perhaps even 1-year studies that will give an output or give an indicator that would be useful for elimination and hopefully will help the program in their efforts. Alongside not dismissing the really serious impact that basic research has on all infectious diseases, we've also made an effort to integrate across the ICMR institutes the thematic research that has to go into malaria, but in terms of a basic research component. And there are lots of issues here, for example, discovery of new insecticides, discovery of new antiparasitic molecules, discovery of vaccines, and of course, epidemiology, correlations with malnutrition, hemoglobinopathies, etc. So there are a huge host of studies that have a very critical basic research component to them that must be carried forward in parallel to the elimination studies. And therefore, this IMM has been launched, and we are very much hoping that partners from non-ICMR institutes will also be able to participate in these calls, which will come out soon. So keep, we'll let you know as and when that happens. So by nature, the IMM studies or ICMR's malaria mission studies are multi-PI, multi-centric, and multidisciplinary. And the whole idea is to really foster cross talks between independent PIs who are very strong, who have an interest in malaria, who may not necessarily be interested in elimination research, but are interested in basic research. But by virtue of bringing together different facets of malaria research, we then are able to produce a strengthened program, which has a strong basic component, but eventually, of course, a translational component. So we are quite excited about this theme that we've launched. And some of the ideas that have been floating around, around which both MERA and IMM are going to be built, are mapped here. And these are just few of the ideas that have been floating around. There are lots, and we are of course very open to any new ideas, and there are lots more going around. But if you start from the bottom, just for the ease of this presentation, smart epidemiology. Now I'll show you some examples of that. We've made some strides in that direction. If you skip to number 8, Dr. Utpal Tatu, my good friend and one of the top omics person in India, will be giving a talk later on, and we are really hoping to pull in and bring in expertise from outside in order to bring in the tools of omics to bear new light in terms of insecticide resistance, drug resistance, markers, etc. If you skip again, I'm skipping just because most of you know these very well, and I don't want to dwell on them for too long. But Vivax, as you know, and I'll show you later, remains a problem and may become a more dominant problem within the scale of reducing malaria, because it's sort of considered harder to eliminate. And the issues of relapse with Vivax, G6PD deficiencies, etc., remain very much as they were 20 to 30 years ago. And I think there is a scope for that in terms of basic research where NIMR and its partners within the ICMR institutes and outside as well can contribute. And again, I'll skip, but we'll skip to point number 2, vector biology. Of course, I'll show you examples where, as I mentioned earlier, there is a huge amount to be learned. I'm not a vector biologist. I'm myself learning at NIMR, but I understand from the data that we've seen so far that we are in good balance for the moment in terms of IRS and alliance, but that can be tipped over given the propensity for both the parasite and the vector to mutate rapidly under stressful conditions. And so we have to be watchful there as well. I'll show you now some of the examples of some of the work that we've done over the past year or so, just to give you a flavor or taste or glimpse of the activities that we are trying to integrate. Now, by themselves, these are not complete, but I think they're sort of very encouraging and show you the direction we are going in. So in Kheda district of Gujarat, we've had a malaria clinic running. In fact, it was the first field unit of NIMR of the erstwhile MRC set up in the 80s. And we've actually managed to digitize all the data, epidemiological data. And you can see all of that is actually mapped onto this one graph. And it's really sort of comforting to see. You can see, for example, from 2005 onwards, how there's been a significant dip in the malaria incidence in Kheda districts. Now, these are walk-ins into the malaria clinic that NIMR runs. And above that, you can see the vector and biological control tools, parasite control tools that were used. For example, the introduction of ACTs under the National Drug Policy, introduction of pyrimethines, L-alliance, etc., and biological control of breeding habitats, etc. And they have really reduced the incidence of malaria here significantly up till 2019, you can see. But you can also see, of course, here that perhaps there has been a greater decrease in P. falciparum than in Vivax, as we had suspected. And Vivax, of course, remains a problem in this area. Now, we looked further at these data, and it really goes to show how vital it is to have granular level data, month-wise or even better. But when we looked at these data month-wise, we saw an interesting trend. Bear with me, you just have to look at the bottom two circles on the left and right. On the left-hand side is the P-Vivax clinic data that we have, and on the right-hand side is the program data. There is a national program, Dr. Abdesh Kumar's program data on Vivax incidence in the last decade or so, let's say 2009 to 2019. And what you can see is in these wheels, monthly wheels are the peaks of malaria cases. And it's very evident that in the PV clinic, for example, there are peaks happening in previous years up till May. And in fact, if you look at the middle circle, you'll see there was a peak in May as well as in August. So there is a pre-monsoon peak of Vivax incidence, and there is a post during monsoon scene of peak as well. But that has not greatly reflected in the program data. If you look at the right-hand side wheel, the bottom wheel, you can see that the program data peaks are all clustering towards September. The second peak is lost somehow, which actually happens because of paddy plantations that start in March or so, and there is a lot of water hanging around, and so you do get a second peak in Kheda district. So looking at these sort of data month-wise really is very revealing, because you can immediately make out that there are twin peaks and there are really two peak seasons to worry about in terms of incidence in this particular one district alone, Kheda. And we won't go into this. This paper is under review, but there are lots of other analyses we've done in this paper that are similar and show how valuable granular data can be when you're looking at them in this context. I'll jump guns to, that was actually the previous work was done by Dr. Bharia, who's an officer in charge of Kheda district, of our unit in Kheda district. This particular work has been done by Raju Ranja, who's officer in charge at Raipur. And he's interested in forest malaria. And you can see here, this is a very interesting map. This is basically the usage of, the level of forest areas across India, and the APIs are mapped here, API 2019. And you can see the largest red dot corresponds to APIs between, well, over 5 up till whatever the maximum number is. And you can see there is definitely a clustering of malarious regions with forest areas. This is, of course, not rocket science. This is evident that this should be the case. But the problem is the accessibility in these areas and how to cover forest malaria going forward, because these areas are hard to reach in general, and there are issues about accessing them in any case for various other reasons. If you look at table 1, there you can see that the forested areas, the APIs are really quite large in the forested areas, limited areas that I'm showing you in this particular table. If you look towards the northeastern states, you can see there's a clustering again around Tripura. All the forested regions and the APIs are quite large. And so there is, of course, a strong correlation between malaria and forested areas in India. That's also evident here. On the left-hand side top panel, you can see of the bars, the red bar shows others, and the forested areas are in blue. And if you just look at the number of deaths in 2019, the forested areas provided much more death data than or rather there were more deaths in the forested areas than other parts of India. Of course, therefore highlighting the fact that forested areas remain a foci for serious malaria. And what's shown on the right-hand side is also revealing. You can see that the incidence of P. falciparum is in fact more or less constant or even increasing in the forested areas, whereas it's somewhat dipping over the past few years in non-forested areas. So forest malaria remains a crucial problem in India, and I think we'll have to come up with new strategies to cover for that. A few other interesting notes on this. I know we have partners, international partners in this meeting who have been championing the understanding of artemisinin resistance and of drug resistance in particular. And this is a serious cause of concern, not only worldwide, but of course in India as well, where artemisinin resistance, to my knowledge, hasn't been really validated or documented to the extent that it's believable, but one never knows. And so what I'm showing you here is the potential to import mutant parasites from across our border, from Myanmar and further east, for example. Many of our states shown here line up with Myanmar, and also Mizoram and Tripura are both exposed not only to Myanmar, but also Mizoram is exposed to Bangladesh, and Tripura is also exposed quite significantly to Bangladesh. And so the parasites can actually be imported from these countries who are mutant parasites. Equally, we can actually export malaria to Bhutan, and what's shown here, and Bhutan is very keen on elimination. They're nearing elimination, and we don't want to be a nuisance to our neighbors either. So you can see the 10 odd districts that sort of circle Bhutan or semicircle Bhutan here. Some of them happen to be malarias, and I think so. Therefore, there are issues about our, in context of malaria elimination and malaria control, our responsibilities to our own citizens, but of course also our responsibilities to our neighboring citizens, because the problem can, what goes around comes around. And so we have to be very cautious going forward in these areas as well.
Please do it quickly because we are running out of time.
Okay, sure, Sachin. So here's, I'll try to be quick, but I might still need 5 minutes. Here's, we are interested also in looking at the landscape of all 5 plasmodia that cause human infections. And what's shown in this little graphical representation is the fact that detection limit with PCR is really the optimum way to get parasite rate in the populations, no matter what particular parasite you're looking at, especially in the context of elimination. RDTs or microscopies by definition will miss significant amount of subpatent and low density infections, which therefore are a cul-de-sac in terms of the road that goes towards elimination. And we've initiated some studies in this field. This is just a paper that we brought out last year, where we did a prevalence study of all the literature that's ever been published on the 5 all 5 human malaria parasites, and you can see at the India's map E and F, one is a decade from 2000 to 2019, the other is 2010 to 19. And you can see that wherever one is starting to look for the other non-PF, non-PV species, one is able to find them. And so you need to be reliant on molecular tools to be able to come up with these kind of epidemiological surveys, where clearly the parasites are not of interest, are not just PF and PV, but the other 3 as well. Very briefly, we've conducted, NIMR has conducted and already shared the data with the program on the prevalence of insecticide resistance. This study was done in 15 states across India. I won't belabor this point, but of course, we need to be cautious going forward in terms of watching resistance in all primary vectors and even in some secondary vectors. But specifically of the 15 states we looked at, you can see the distribution for culicifaces. The resistance to DDT is 50 in 50 out of 52 districts, to malathion 27 out of 44, and to deltamethrin 17 out of 44. This is not quite alarming as yet, but that doesn't mean that we actually take our eyes off insecticide resistance. As I told you earlier, and all of you know already, it's one of the vital tools for malaria control. And so some of the other avenues that we are considering, in fact, and we would really like for this to be opened up for discussion in case people are interested in this later on, is we are wondering whether India can consider integration of the 3 eliminable diseases that are under the program. This is basically for malaria, lymphatic filariasis, and leishmania. And this is an interesting study that's already published, where we show that there are a huge number of districts, upwards of perhaps close to 100 districts, which have coexistence of all 3, or at the minimum of 2 of these 3. And so perhaps there's some level of integration can happen in terms of efforts to control this. We are also wondering why empowerment of the communities can't be done by allowing rapid diagnostics to be available in the free market. Same is true for LLINs. We've developed a very nice fever tracker app, which is runs on Android, and this is going to be a very nice surveillance tool, which we will be very happy to share with the program. And lastly, I'm going to end up with this. we've actually developed a very nice malaria dashboard that integrates all malaria surveillance data and is very user-friendly. And I'm just going to flip through some of the slides and you'll see how powerful this malaria dashboard is. So this idea basically was born by a couple of papers we wrote earlier about how there is a need to integrate all available malaria data in terms of, all spanning from basic biology to epidemiology at a digital dashboard, which is then available not only to the program people, but also to basic scientists who want to develop questions around the epidemiology of malaria. And so one of these have sort of worked out quite well already, and I'll show you here. This is all thanks to Dr. C.P. Yadav, who I hope is listening. He's a scientist at NIMR. And also I would like to specifically request Dr. Avdesh Kumar of the program to see this dashboard. So this dashboard is extremely flexible for looking at epidemiology data. In this case, we are showing you, for example, if you were interested in Changlang district from Arunachal Pradesh and its API over the past 20 odd years, you can actually trivially map this instead of going through tons and tons of Excel files to get these numbers out. If you wanted to look at districts within Assam and the distribution of API over just 1 year, in this case 2000, or over a time series, you could do this trivially. If you wanted to look at the same districts of Assam and you were wondering what is the spread of the APIs in these districts? What is the median and the means of these APIs? Is it really getting better, or is it sort of a yearly thing when in some cases it gets worse and so on? You can actually do that here as well. If you were to ask the question between districts comparison on the basis of API, you could actually select out what's shown on the left-hand side, the years that you're interested in, the API, which is one of the indicators, the particular state Assam, and the districts you are interested in. And so you can very easily and very quickly zero in onto the districts which are problematic. On the other hand, the districts which are behaving well in terms of malaria control. If you had a question, for example, what is the percentage change in malaria in various districts in a given year, for example? Are things really getting better between a year or two? For example, you could have an outbreak and there was really not of a problem till a few years ago, but suddenly there's an outbreak. You'd be able to trivially, again, be able to analyze those data here. And lastly here, this is also very nice because you can see here, you can watch the varying distributions of P. falciparum versus P. vivax infections district-wise. And so in some cases, you can see that there isn't much difference in the proportions of the two. In some cases, you can see that Vivax is beginning to dominate the total malaria, and in other cases, contrary to that. So this is very nice because the general wisdom and understanding is that the P. falciparum areas under control and elimination will relent, but Vivax will hang around for a longer time. It will be difficult to, or slightly more difficult to eliminate. And so this is a very trivial, but a very easy way to then zero in onto the districts where you can actually see that trend and follow it. I think I'll end there. Thank you. And I apologize for going over the time by a few minutes, but I really wanted to sort of give you a glimpse of some of the things that we've been doing. And over to you, Sachin. Thank you.
Yeah, thank you. Thank you very much, Dr. Amit Sharma. And thank you very much for showing the efforts, research efforts, what NMR is doing towards malaria elimination. And so we'll move to the next speaker. And sorry, sir, for the limited time. And I'm sure you will not cut water supplies to my office for this.
Yes.
Now we'll move to Dr. Bart Knowles. Dr. Bart Knowles is a PhD MBA, is a medical public health entomologist, a science manager, university lecturer, and with a broad interest and experience in development issues, the environment, global and public health, infectious disease management, with a special interest in the research and training on the biology, behavioral ecology, integrated control vectors of mosquito-borne diseases, especially malaria and dengue. Yeah, over to you, Bart.
Thank you very much. Yeah, let me start off by just saying that currently I'm actually based in the Maldives. I'm not in Africa, where I used to work a lot on malaria. So when I talk about my malaria experiences, I mostly talk about Africa. Currently, I'm in the Maldives trying to free islands of the presence of Asian tiger mosquitoes, which obviously are not transmitting malaria, but arboviral diseases. Anyway, before I continue, let me start by thanking the organizers of this symposium for inviting me to speak to you today about a topic that is actually very close to my heart, and that's malaria elimination, if not malaria eradication. Okay, the difference between the two, as you probably know, is that elimination is a sort of on a local or a regional scale, the absence of any transmission, any people carrying parasites, whereas eradication is sort of the disappearance of the pathogen from the face of the earth, from the planet. We have eliminated smallpox as the first one, eradicated that from the face of the earth, and we are close to getting rid of guinea worm in Africa, and of course polio, which is very close to being eradicated, with only a few pockets of wild-type virus circulating in Pakistan and Afghanistan. So when we talk about malaria, it's the interesting issue, and it was already mentioned before, is like, how do we close this gap between what we're doing at present and how we actually move down to complete zero and keep it there? And agendas for malaria eradication, for the global malaria eradication, have been drawn up and are freely available online. I mean, you can find these images as to how the malaria map will shrink over the years to come. And documents and reports have been circulated, like the WHO Global Technical Strategy for Malaria 2016-2030. And of course, every year in December, we look forward to seeing the World Malaria Report being published by WHO, which basically shows us where we're at in terms of the global malaria eradication efforts. As the founder of Malaria World, which is an online platform that publishes weekly newsletters with all the latest publications on malaria, with meeting announcements, etc., with close to 11,000 members today in more than 140 countries. We follow here very closely what is happening in terms of the global efforts towards elimination. And today I want to tickle you with 5 different points where I believe that we need to start doing things differently or better. So unfortunately, in spite of the initial dramatic successes that we've seen since the turn of the millennium, with the exception now of India and the slides that we just saw, how there's been this remarkable decline in the number of cases across this large country. Overall, if we're talking at a global scale, then things have pretty much stalled over the last 5 years. We have not been making any dramatic progress in the last 5 years, which of course is deeply worrying. And actually the global technical strategy as it is, it's up for being revised. It's going to be rewritten. So we are seriously off track. And the question one may pose, of course, is why are we so seriously off track? Now, COVID-19 obviously is part of that discussion, but that's basically just the last year. So the 4 years before that, we also were sort of stalling in terms of progress. And the question is, why is that? Now, I'm not an historian, but I do like from time to time to use quotes from famous people to try and get some points across. And the first quote I really like is a quote by Albert Einstein, which says, if you do what you did, you will get what you got. And basically what this means is that if you believe that you can solve your problem, in our case, the malaria problem, with the same tools and the same approaches you've used before, that you will get the same outcome. And in our case, that outcome is stagnation. Okay, so no more progress towards malaria eradication if we continue to do what we have been doing over the last 2 decades. So change, therefore, is needed. So not more of the same. A second quote of Einstein that I really like is this one. We cannot solve our problems with the same thinking we used when we created those problems. In other words, if we believe that, for instance, malaria drug resistance can be solved with just another deep mining exercise of chemical compound libraries so that we can discover a new chemical with anti-Plasmodium properties, turn that into a drug, put it on the market, it can be almost guaranteed that we will face the same problems later on as we are facing today, drug resistance. So what happened in the Thai-Cambodia border in 1957, when for the first time we observed resistance to chloroquine? At that time, it could have been predicted that we were going to be facing the same problems with the artemisinin monotherapies in exactly the same region, the Thai-Cambodia border, half a century later. And then if we lose efficacy against the monotherapy, we tend to think that a combination therapy where we combine 2 active ingredients can then do the job. Well, it won't, or at best, it will do so only temporarily, because it still remains the same problem that we try and tackle in the same way. So with the resistant threat that is building up against the ACTs, the combination therapies, we are now even looking at triple combination therapies, the TACTs. But we still do what we did before, and therefore we will end up with the same result. When it gets to vector control tools, I often resort to a famous quote from Henry Maslow, who said, if the only tool you have is a hammer, you treat every problem as if it were a nail. In vector control, we have been made to believe over the last 5 decades that chemical interventions are the sole interventions with which we can score real impact. And in spite of losing the game again and again, chemical companies make us believe that new insecticides for indoor residual spraying and for bed net impregnation are the only way forward to solve our problems with insecticide resistance. In turn, as a global malaria community, we continue to believe that this is actually the case. The Gates Foundation has poured hundreds of millions of dollars into finding new active ingredients for insecticides, only for us all to know that once these chemicals hit the market and are being used on a large scale against vectors, that within no time we're going to end up with the same resistance issues as we are facing today. Not only because we're using these chemicals in vector control, but also because the chemical companies want to market these also against agricultural pests, which is a much bigger market than the vector control market and is actually promoting the development of resistance in our vectors. So the question why we are stalling, why we're not making new progress, can be answered pretty much by looking at the way we address current challenges in terms of drug and insecticide resistance. That's not the whole story, but it's certainly a major contributor to the story. So we tackle these problems with the same old solutions, merely to find ourselves addressing these exact same problems a few years later. So now that chlorfenapyr and clothianidin, both belonging to new classes of insecticides, one of which is actually banned in the EU because of killing bees, have come along, we will see resistance surfacing, let's say, within the next 5 years. So the question then becomes, why do we actually stay in this treadmill? Why do we keep on doing the same thing, ending up with the same problems? Malaria control and now malaria elimination has pretty much become a commodity-based industry. Our weapons consist of rapid diagnostics, antimalarial drugs, bat nets, and insecticidal sprays. And there are hopefully some vaccines on the horizon. Actually, just recently, there was new hope from the phase 2B trial with the R21 vaccine developed in Oxford. So that's good news. But they're still commodities. It's commodities based. And the general thinking is that if we maximize coverage with these commodities, that we will knock malaria over the head. Or asked in a different way, if we send a large number of cannons, rifles, bullets, and machine guns to the front lines, will that result in us winning the war? And I would say the answer to that is no. So merely jacking up the provision of commodities is therefore not going to help us to bring down malaria further. And the reason for that is that at present, there is no strategic application of multiple tools in an integrated fashion, adapted to local agroecological settings, vectorial systems, and transmission dynamics. So all we have focused on, or primarily have focused on, certainly in the African context, is primarily bat nets without any further regard for additional tools to drive transmission down to zero. So in essence, we are fighting a war with commodities, mostly one, but we lack the generals that can help us define successful strategies on how to deploy these tools to reach zero cases, 0 deaths, and then keeping it at 0. And perhaps this is also the major reason why we are facing all these problems with resistance. The longer we apply drugs, the longer we have to apply insecticides. When we control malaria rather than eliminate it, the more we give the parasites and the vectors a chance to build up resistance and let evolution do its work. Invariably, wherever we have to keep the fight going on at the level of control, evolution will catch up with us, putting us back to square one, back to the drawing board, looking for new active ingredients for drugs and insecticides. So I guess the message then becomes, we need to learn how to develop integrated packages that are suitable to drive down malaria to zero and actually keep it there. And funnily enough, if we look at the more than 100 countries worldwide that have successfully already eliminated their malaria, we see that almost without exception, these countries did exactly that. They integrated a set of tools, adapted these to local circumstances, and were rigorous, almost militaristic in style when it came to weeding out the last few and final cases from a human population. Today, in sub-Saharan Africa, as far as I know, there is no single place where we have succeeded in doing just that, or even are trying to do this at present. Also, we seem to have forgotten that these successes, so the fact that more than 100 countries already successfully eliminated malaria, were accomplished without a malaria vaccine, without insecticides, without genetically engineered mosquitoes, without attractive toxic sugar baits, without spatial repellents, and without a whole other suite of new tools currently under development, of which we are made to believe that without these new tools, these novel tools, we will not be able to succeed in eliminating malaria. And that is simply not correct. Of course, it will be great to have more tools in our toolbox, don't get me wrong, but we have come a very long way without them. India itself, and it's been said before, India itself was massively successful in the late 1950s when it launched, as part of the global malaria eradication campaign, the largest public health campaign in the history of mankind. Up to 1 million people were involved in this campaign, which largely relied on indoor residual spraying with DDT. Where in the late 1940s, malaria cases could be as high as up to 70 million per year. Just 3 years into the campaign in 1961, this had been driven down to just some 50,000 cases for the entire country. India, therefore, almost eliminated its malaria in the early 1960s. Equally remarkable was the story from Sri Lanka in those days. They embarked on a similar campaign, and although they had 2.5 million cases per year, after a few years of this intense campaign, had less than 20 cases of malaria in the country. Nevertheless, both countries failed to drive down transmission to zero and failed to complete the last mile. And failing to complete the last mile brings me to my next point. Our over-reliance on communities to eliminate malaria. There is, as far as I know, not a single country that has eliminated malaria based on community engagement only. And the word only is an important word in this regard. Obviously, nowhere should any intervention be staged without community consent. That is, community members agreeing to an intervention being rolled out in their surroundings. And if people then wish to be enrolled and participate in the actual work, then obviously this is desirable and is fine. However, if we are to rely on those same communities to walk the last mile with us, then I believe that we're naive. How can we build on and depend upon often impoverished communities to help us eliminate malaria if they hardly see anyone in their surroundings suffering from the disease, if it's practically gone? People have limited time and they will set their priorities accordingly. So if malaria is almost gone, communities will lose interest. And I think what is happening with COVID-19 here in the Western world is a perfect example of this. As soon when cases started going down, sort of when we had our first wave, politicians and communities, all of them, they reduced the stringency of the measures that we were taking to control transmission of COVID. People didn't want to wear their face masks anymore. People considered it gone. And this merely resulted in the next wave of cases coming along. And we're right in the middle of that here in the Netherlands. So walking the last mile in malaria is the hardest and probably the most resource intensive mile, both in terms of people as well as in terms of funding. And I believe that we will have to walk that mile for communities, like we did in the Western world when eliminating malaria was so successfully done decades ago. In other words, only through vertically staged intensive monitoring and evaluation campaigns will we be able to sieve out the lost parasites from communities. And this will be very hard, but I believe that we don't have a choice. Now, a great example that I want to give you is what happened in West Africa in March 1930, when in the harbor of Dakar in Senegal, there was a French marine ship that was bound for Natal in Brazil to cross the Atlantic Ocean. On board of that ship, the African malaria mosquito Anopheles arabiensis, which occurs in the gray area across the continent, was found. And this mosquito hitchhiked across the Atlantic Ocean and settled upon arrival in Brazil. And there, over a time frame of 8 years, it spread over an area of 54,000 square kilometers, a large area. So this invasion of this African malaria mosquito was remarkable because they were superb vectors of South American Plasmodium falciparum. Dissection of mosquito guts showed an astonishing oocyst rate of 63% of the mosquitoes. And dissection of the salivary glands showed a sporozoite rate that was about 30%, which is massive. This vector competence has not been found anywhere in Africa. So the person who got really concerned with this invasion was an American epidemiologist called Fred Soper, seen here in this picture, who lobbied hard with the Rockefeller Foundation in New York to basically give him unlimited funds to try and eliminate this invasion of this African mosquito. By 1938, when the invasion had entered the Jaguaribe Valley here on the left, an epidemic struck which affected more than 200,000 Brazilians, and some 15,000 people perished due to the lack of antimalarial drugs. And then the interesting thought, Soper got to work in 1939, and he trained some 4,000 Brazilians to do nothing more than to locate standing sunlit water where this mosquito was breeding and treat that water with Paris green, an arsenic salt. And then, remarkably, it just took 18 months to finish the job. Within 1.5 years, this entire area of 54,000 square kilometers was freed of all African malaria mosquitoes. Soper focused on larval control, which, since the malaria models of the 1950s, became less popular when the focus shifted to reducing the daily survival of the adult female mosquito. That's why we do indoor residual spraying, and that's why we have LLANs. Nevertheless, it has to be noted that not a single country in the world actually has eliminated its malaria without larval control, without larval source management. And although as scientists we are inclined to look for new things rather than the old and the tested, we ignore the power of that success that was done in Brazil. In essence, this campaign delivered the template of how the invasion of the Indian Anopheles Stevens eye in the Horn of Africa, which we are witnessing today, could and should successfully be managed. These 54,000 square kilometers, if I project that on small countries in Africa, there's actually 8 countries in the continent of Africa where children are suffering from malaria and people are dying from malaria every single day that are smaller than or about the same size of the area that Soper freed of African malaria mosquitoes completely some 80 years ago. If we were to double that area for such campaigns, if we would do the same thing that we did in Brazil, but now on double the size, so go to about 100,000 square kilometers, then these countries would enroll. If we would do that 5 times the size, then these countries would enroll. And if we would do it 10 times, we would be looking at major chunks of the continent, staging short duration, massive size campaigns by thousands of people that would use tools for that short duration so that parasites and vectors would not get a chance to develop resistance to the drugs and insecticides that we target them with. Thousands of young people could be trained to do what Soper already did. And looking at unemployment rates in the continent of Africa, such people could be found easily. There's lots of people that could be trained to do exactly what we did 80 years ago. And I even believe that we could do this much better. Soper went about on mules. He was drawing his maps by hand, and he used a very toxic larvicide, Paris green. These days, we have massively improved infrastructure and transport. And we can augment that. We can augment that with modern technology, such as satellite imagery, computers, geographical information systems, the apps that were already spoken about earlier on. And I bet you if Soper was alive today, I'm sure that he would be out there eliminating malaria and not studying it. And for those doubting it, Soper was also the genius that eliminated malaria from the first African country, from Egypt, between 1943 and 1945, doing the same thing, training 2,000 Egyptians to run a massive campaign. And yes, that was done during World War II. I could talk about this for a long time, but I have to wind up and summarize the 5 key points that I want to give you in this talk. The first point is that we need to move towards tailored, integrated campaigns, like the forest malaria that was just spoken about, tailored packages specifically for forest malaria, rather than focusing on a limited number of tools that we just blanket across continents. Second, we should move away from delivering commodities and then assume that malaria will disappear. We need to strategize much better when elimination is the goal. Third, I do question the role of communities beyond giving consent when elimination, not control, when elimination is the goal. When a problem is almost gone, people shift their focus to more pressing problems. Fourth, it is time that we acknowledge the role of larval source management. And it is unlikely that we will succeed in any other country to eliminate malaria without incorporating this tool in our package. And finally, it is worthwhile to learn from past successes, as I've just shown you from Brazil, and see how our today's advanced knowledge and our new technologies can make such campaigns even more successful today. I thank you for your attention.
Thank you very much, Bart, for the excellent presentation and making some good points on, the new tools, new strategies, and vector control by engaging community. So thank you very much for your time, and looking forward to listen more in the panel discussion.
My pleasure.
Thank you. So next, may I request Professor Utpal Tatu? Professor Utpal?
Yes, I'm here.
Yeah.
Hello, Dr. Sharma.
Hi.
Okay.
So Professor Utpal Tatu is a professor at Indian Institute of Science, Bangalore, and his research focus on the neglected infectious diseases with emphasis on developing better methods of diagnosis and treatment in the area of health. Dr. Tatu is a recipient of national and international awards, and he is an elected fellow of Indian Academy of Sciences and a founding member and former president of Proteomic Society of India. Yeah, over to you, Dr. Tatu.
Thank you, Dr. Sharma. Thank you for for inviting me to be part of this conference organized by NIMR. I'd like to thank the director NIMR, Dr. Amit Sharma, also for this wonderful initiative and a feast of wonderful talks by eminent people in the field. I enjoyed the earlier talk very much, and I look forward to the rest of the talks. I'm going to talk to you today about the aspect of malaria, which I think is going to be the big challenge in the malaria elimination program that we are all looking forward to and what we are participating in. This is, I think, going to be a part of a challenge that we'll have to understand. And again, a lot of molecular work will have to happen in this particular area of understanding the sexual stage development of malaria, I think. And that's what I'm going to do really, is to share with you some basic science research that I'm trying to do and over the last few years in this quest to examine how does the parasite switch to the sexual stages? So yes, I'm going to show you some basic science data involving omic sciences and some of the clues that we have found that might help us understand how the parasite regulates sexual stage transmission. Just to introduce myself very briefly in terms of what my lab does, malaria was indeed my first love. I started my research career in '97 on malaria and have continued working on malaria. But over the years, we have also incorporated related themes in my lab. Related themes, other infectious diseases, some of which are shown here, a host of protozoan infectious diseases, some of the fungal pathogens such as Candida, and more recently we have embarked on viral infections, particularly COVID-19 and rabies, in trying to understand. These are both diseases which are obviously relevant now, but also have been relevant traditionally, especially rabies, and that's really what we are trying to do. So over the years, we've used experiences of our work on malaria to understand many of these other infections. And oftentimes our focus has been on a particular pathway, namely heat shock response pathway. How do these parasites respond to stress? How do they cope with stress? And is there a possibility that the ability of these parasites and infectious agents to respond to stress could be the Achilles heel and could be the soft target for us to find better therapeutics? That's really been the philosophy of my group, and we've done a bit of science in this area. The reason also, especially in case of protozoan infectious diseases, we have chosen to work on some of these pathogens is because organisms like Giardia, Entamoeba, Trichomonas often actually share facets of course, eukaryotic cells, but also have some sort of remnants of what might be, reminiscent of prokaryotic biology. And so these are the early branching eukaryotes, as they are often called, and there are some interesting secrets about what may have happened during the time that eukaryotes evolved during the evolutionary periods. With this broader theme, then I'm going to tell you a little bit about this new area that we are pursuing in my laboratory, and what are the tools that we are applying, and very little of what we have found. Indeed, I must confess that we have really not made much progress. We have not published a lot of papers on this particular aspect, but yes, we have lots of ideas. We really have some clues, and I think what's very clear is that this continues to be the least understood aspect of malaria biology. So very quickly, I'll go through the three main points in my talk. A brief introductory aspect about my malaria work, bit about how we've applied the tools of metabolomics in addressing this question of gametocytogenesis, and then what have we learned in the last couple of years that we've been working on this. Just to remind you, traditionally, for the last couple of decades, my laboratory predominantly focused on heat shock response pathway, delineating how the parasite, malarial parasite, during its asexual stage development, uses this chaperones or heat shock proteins to combat environmental challenges, and particularly aspects of febrile episodes that are experienced in the patient. And over the years, then we've characterized a host of important and oftentimes essential heat shock proteins and their complexes and the pathways that actually regulate their expression, including accessories of their transcription factors and epigenetic modifications that regulate their expression. And what we find really is that environmental cues are in fact pursued by the regulatory complexes comprising of essential heat shock proteins such as HSP90. Most of you in India, of course, are aware that that's really been the main emphasis of my work, heat shock protein biology, and this was predominantly focused on the cytoplasmic pathways, the cytoplasmic heat shock proteins which perceive stress, and then they oftentimes the transcription factors that regulate their expression are bound to heat shock protein 90, and thereby the role of HSP90 in regulating not only the environmental stress responses, but also how the parasite grows and develops. An interesting corollary of this work was that there was a potential of looking at this stress response pathway as targets to combat malaria, and there was some interest shown. And in fact, other people in the field who went ahead of me and have now developed molecules that specifically target malaria heat shock proteins to possibly explore their role as anti-malarial drugs. What I'm going to talk to you today is really somewhat different. And over the last few years, we have turned our attention to the secretory pathway. So really the endomembrane system of malarial parasite, because today we understand so much more about how malarial parasite deploys its secretory and membrane proteins through the complex secretory pathway that the parasite establishes for itself in the infected erythrocytes. And what I'm going to show you is that some of the components of the secretory pathway are important. And then again, stress to the secretory pathway holds some important secrets about how malarial parasite has evolved to utilize these as a cue in switching its developmental stages. What I'm going to show you is that endoplasmic reticulum and what goes on inside of the endoplasmic reticulum has something to do with parasite's choice to switch to gametocytogenesis. So I'm going to show you that given the sort of the broad map of the malarial parasite inside of the infected erythrocytes, as you can see in this slide, malarial parasite exhibiting its own endomembrane system, the nuclear membrane, endoplasmic reticulum, remnants of Golgi, food vacuole, apicoplast, and then the parasite membrane, followed by vacuole, and then parasitophorous vacuolar membrane, and then the host cell membrane. Fascinatingly enough, malarial parasite has developed capacity to deploy proteins to all of these locations during its asexual development pathway. And in addition, it also deploys protein outside of the host cell. It has the capacity to secrete proteins out and also lodge proteins on the surface of the infected erythrocytes, which often serve as important pathogenesis tools for it to establish in the host. Given that the parasite relies so heavily on a secretory pathway for its pathogenesis, it came as a surprise early on when we found that it is ill-equipped actually to make the secretory pathway robust. What we found was some of the key proteins found in the endoplasmic reticulum, including protein disulfide isomerases and its close accessory proteins, were actually deficient. While PDI is present, but if you looked at the number of homologs of PDI, the isoforms of PDI, peroxyredoxins were ill-represented in the genome of malarial parasite. And then again, the biggest sort of finding that we made was malarial parasite was ill-equipped to cope with redox stress, or what is often called as the unfolded protein response in higher eukaryotes. And this is what I'm going to show you in the next few minutes, focusing predominantly on how our analysis of the secretory pathway of malarial parasite gave clues about what the does to regulate a secretory pathway. Now, just to remind you all, the eukaryotic biologist, that the Nobel Prize winning work by Peter Walter showed that unfolded protein response, a mechanism that most eukaryotics, in fact, he claimed that all eukaryotic cells are endowed with UPR pathway, wherein accumulation of unfolded proteins in the endoplasmic reticulum triggers signaling pathways that communicate with the nucleus and thereby upregulate expression of key ER chaperones to help solve accumulation of unfolded proteins in the endoplasmic reticulum. This was phenomenal, where they found actually a cytoplasmic splicing mechanism as a part of the signal transduction process. We noticed during our efforts to understand secretory pathway of malarial parasite that some of the key components of seemingly universally present UPR pathway were glaringly missing in Plasmodium falciparum. And that's really where our journey in terms of understanding stress to this to the secretory pathway of malarial parasite began. We observed that IRE1 a membrane protein, which is known to actually generate a transcription factor upon complex splicing in the cytoplasm, actually results in a transcription factor that upregulates expression of ER chaperones in the endoplasmic reticulum, was completely missing in Plasmodium falciparum. In addition, ATF6, a Golgi localized protein which is involved in upregulating UPR, was missing. But then some of the kinase proteins, which are the bare minimum, minimalistic UPR components, seem to be present in the malarial parasite. But the most important components of ID1 and ATF6 were glaringly missing in malarial parasite. And that's when we began to address, how does the parasite cope with ER stress? So as often as cell biologists often do, they expose cells to conditions that will upregulate unfolded protein response in the ER, such as reducing agents and oxidizing agents, which sort of mimic stress to the ER, which is what we did. And we found that upon exposure of asexual stages of malarial parasite to reducing agents, which could be which are known to trigger unfolded protein response in other higher eukaryotes, actually gave rise to some very interesting gene expression patterns as we observed early on. We found that in this asexually growing malarial parasite, in addition to many of the interesting changes that happen upon exposure to ER stress, we notice a minority of sexually expressed genes, sexual state specific genes that were upregulated. And this is really how we began our sort of a deeper understanding of how this unfolded protein response of malarial parasite may be connected to gametocytogenesis. To cut the long story short, one of the students in the lab at that time noticed, Shweta Chaube, she noticed that among the plethora of genes that were up and down regulated, a few of them stood out because they were usually expressed only under sexual stages of malarial parasite. And then on, she went on to confirm this proteomically using differential gel electrophoresis, wherein you would use Cy3 and Cy5 labeled protein extracts from different cells and mix them together and look for genes that are upregulated, shown by red, gene products that are upregulated, shown by red, or green, downregulated, or those which are same as by yellow. And using these approaches, we confirm proteomically what was actually originally observed transcriptomically, that the genes corresponding to to sexual stage transition were upregulated, and proteins were correspondingly upregulated in response to changes that would trigger unfolded protein response in other eukaryotic cells. But because UPR pathway was missing, or the key components were missing, the parasite responds in a different way. So really, the first clue was that sexual state specific genes and proteins are expressed. Does this actually mean anything phenotypically? I'm cutting the long story short. I'm really just showing you the clinching experiments here, but to show you here, starting with an asexually growing malarial parasite, upon exposure to redox stressors, variety of them, we found that over a period of time, systematically, they went through all the stages of gametocytogenesis, showing you stage 5 banana-shaped gametocytes overexpressed upon exposure to ER stress. And this was, of course, well represented in various, in addition to P. falciparum model, we also showed this in vivo in P. burgei model. What this meant was the reflection of gametocytogenesis was upregulated. There were fold changes that were happening in upon exposure to redox stressors. Now, this is where I want to just emphasize a little bit about how little do we understand about this particular aspect of malaria biology. We know so much more about the asexual stages and the gene regulation, the proteomics, and so on. But how do a few parasites, among the few thousands that are growing, or maybe few, large numbers that are growing in the bloodstream, decide to switch to gametocytes? Even today, I think this is actually only a partly understood process. Only a small number of asexually growing cells choose to then turn into gametocytes. And that's something is a big open question even as of today. What I'm going to show you in the rest of my talk really is the early results that I shared with you indicated that when you provide stress to the endoplasmic reticulum of asexually growing parasites, you would find some of them turning into gametocytes. And the full upregulation of gametocytes indicated that stress to the endoplasmic reticulum could be one way, one sort of a cue that parasite uses to change into gametocytes, into sexual stages. So just to recap, I think transcriptional upregulation data, which of course I only had chance to passingly show you, proteomic data, and then the rest of my talk will focus then on metabolic validations of some of the findings that we had, which indicated that ER stress indeed results in switching of asexually growing parasites to gametocytogenesis. Now, as some of you remember, there are various different ways by which we malarial biologists, can turn asexual cultures into gametocytes. There are a variety of tricks which have actually come over the period of large number of years, either nutrient deprivation, exposure to anti-malarial agents such as chloroquine, or exposure just the RBC lysates. These are some of the common ways by which you can in vitro generate gametocytes. But really, the knowledge has been very not really progressed much at molecular level. But today, we of course know the transcription factors which are involved in upregulating gametocytogenesis. But what is the proximal cue? What is the first trigger that converts those few cells to choose to enter sexual pathway? I think, to my knowledge, it is still an open question. In this scenario, it was interesting for us to find that really ER stressors could be one such mechanism by which you could make this happen. But more importantly, we had a molecule. We had a molecule that we could use instead of broader agents such as nutrient deprivation or chloroquine and so on, but a molecule such as a reducing agent that could mimic this kind of trigger mechanisms in vitro. What I'm going to show you then is the answer to the question that what is playing the role of ER stress inducer in a normal malarial parasite life cycle. And this is where metabolomics came handy to us. We had some knowledge about how to use metabolomics as a tool to study malarial parasite. We used a QTOF-based mass spectrometer and MPP software to do global metabolomics. I'm going to skip the details, but also together with Orbitraps and GCMS and LCMS system, we actually narrowed down to what are the metabolic changes happening in the malarial parasite as it grows through asexual pathway. Professor Gordon Langley from Paris, France, was a close collaborator, and he was the one who triggered us to get into this wonderful journey of metabolomics. And I really cherish the association I've had with Professor Gordon Langley, his open-minded thinking, and it really helped us to think of something different from what we were doing in my laboratory up until then. To cut the long story short, we had, again, a heat map representing metabolites that change during asexual pathway of malarial parasite, going from ring to trophozoite to schizoid. And then again, in response to stressors, what are the metabolic changes happening, not only in the secreted, in the secretor or in the extracellular space, but inside the cytoplasm of the infected erythrocyte, and then also within the parasites. I will not have time to show you all the data, but we had very good volcano plots and the principal component analysis, very extensive statistical analysis, which resulted in very precise points in the metabolic pathways that were changing as the parasite grew inside of infected erythrocytes. In the last few minutes that I have, I'm going to show you then that these are the molecules that we are focusing on, that as compared to the control RBCs, which received similar environmental conditions, where we monitored some of these molecules, be it in the redox pathway, be it cysteine, glutathione in its reduced and oxidized state, and homocysteine, we found that some of these redox metabolites were overrepresented. We were fold upregulated as the parasite grew inside of the infected erythrocytes as compared to the control cells. And this was glaring. There were many changes happening, but these were very, very stark changes that we observed. This is sort of a very simplistic representation of the global metabolomics that we did, focusing once again on the redox pathway, particularly the glutathione changes that happen in the redox and reduced and oxidized glutathione levels, ornithines, homocysteines, and that then helped us to make a sort of a working model as to what may be going on when the parasite is switching to the sexual stages. I'll finish my talk in the next 2 to 3 minutes, but to tell you that I think we really had interesting insights into glutathione, which some of you remember is a wonderful tripeptide composed of glutamate, cysteine, and glycine with unusual so-called peptide bond. And I think I'll skip some of the details, but it all sort of focused our attention into the transalpination pathway of malarial parasite. My student at that time, Divya Berry, who's now actually continuing her postdoc, and actually she's with Mohan Narla Das now in that group, continuing actually analysis of the very same work, very sort of similar sort of studies that she started as a PhD student. But to sort of sum up this part, transalpination pathway predominantly is composed of methionins, cysteine, homocysteine, generation of cysteine, and the flux between glutathione in its reduced and oxidized state. We found that homocysteine, cysteine, as well as glutathione were drastically affected in infected erythrocytes. The levels were actually drastically going up. But most importantly, and I don't want to belabor this point too much because of lack of time, we have very extensive statistical analysis of these pathways, and homocysteine is something that we are pursuing currently very quickly. I'm looking at the watch and I feel that I must move fast to tell you that we not only looked at the gamut of redox metabolites, which revealed that as the malarial parasite invades the erythrocyte, it completely shifts the redox status of the erythrocyte towards the oxidizing nature. And this is, of course, a relative shift that you find, and that is something that we were able to quantitate based on the concentration of redox metabolites that we measured. We found that, in fact, this seems to be a common thing among many, many infectious agents, that upon infection of the host cell, they really manipulate the redox pathway. Theme which is commonly seen in virally infected cells, bacterially infected cells, but we found this was also true in malarial parasite, Plasmodium falciparum. But what's the net result of all of this? Here comes the working model that we currently have based on extensive characterization of redox metabolites as the parasite goes through its different stages, and the accessory molecules such as acetinosyl methionine, SAM, SA, and related. I'll skip some of these points, but come down to the final important message that I want to get across. When we looked at the transalpination pathway of malarial parasite, which involves generation of homocysteine, conversion of homocysteine into cystathionin, and followed by conversion to cysteine. These are the two enzymes which are typically present in most eukaryotic cells. What we found, in fact, was that both these enzymes were completely missing in malarial parasite. And thereby, we had a very interesting truncated TS pathway or transalpination pathway, wherein homocysteine is generated and it cycles through SAM, SA, methionine, and so on, but it does not convert into cystathionin and it does not convert into cysteine. And thereby, you have accumulation of homocysteine, as we saw shown in our metabolomics, global metabolomics, and targeted metabolomics data. So we not only had the observation that homocysteine is accumulating in infected erythrocytes, but we had now a genetic confirmation that the reason homocysteine is accumulating is there is no pathway that would bifurcate homocysteine towards generation of cysteine in malarial parasite. And this homocysteine then is actually doing wonderful job. Many of you know that homocysteine is actually a molecule that is notorious for various pathological conditions, but also various regulatory conditions. And here's a single cell, seemingly primitive parasite utilizing this molecule cleverly enough to accumulate it by deleting and possibly willingly deleting two important enzymes that convert it into cysteine, and thereby using it as a time clock, as a clock that indicates its accumulation, indicates the time that it should possibly move on to the next stage in its developmental cycle. So really, this is where the working model is, that we think that the interplay of metabolites in the transalpination pathway has secrets hidden about how the parasite actually excretes out some of these metabolites because of their accumulation, and thereby actually is creating a signal for itself as it goes through its several levels of asexual multiplication cycle, resulting in not only increase in parasites, but also increase in the level of some of these metabolites that has accumulated, serving as a signal for many of its subsequent stages of the development pathway. In the end,
may I request you to summarize, please?
Yeah, this is my last slide. What I'm going to show you really is that metabolomics has helped us to identify some of these molecules. And I'm going to switch and just show you that currently the story is revealing molecular players, in the sense that how this homocysteine actually brings about epigenetic modifications. It brings about histone modifications that regulate gene expression, and around the AP2 transcription factor, there are epigenetic changes that are happen. I'm going to skip a couple of these slides and show you that we have now mass spectrometric evidences showing that histones get homocystinated in malarial parasite exposed to redox stress, and those homocystinated histones are epigenetic marks focused specifically in regions enriched in around the AP2 gene, which is really the master regulator of gametocytogenesis. And with this, my working model, which confirms, in fact, that it's really the metabolite sensing that is one of the early changes, followed by epigenetic modification, and then some of the omic data that I showed you, which actually provides some pieces of the puzzle that that will hopefully in future reveal facets of sexual stage development in malarial parasite. And I've had a couple of projections, but I'm going to end this talk here. I'll just very clearly mention that this theme is actually repeated in many other protozoan parasites, and which is what we are thinking right now. But I want to thank Dr. Divya, who did this main part of the work together with Professor Gordon Langley and many other collaborators. My past students, and particularly Dr. Pragya Acharya, who's now at AIIMS, carrying forward some of the interesting malaria work on HSP40 that she started in when she was in the lab, and then many others who have contributed to this story that I present. And thank you very much, and sorry for exceeding my time, Dr. Sachin. I appreciate your patience.
Thank you very much, Professor Tatu, for a very enthusiastic talk and talk about response pathways. And I'm sure our basic people here, audience, you give different taste of your results, what's going on in your lab. Thank you very much for this.
Thank you, sir. Thank you very much.
So next we'll move to Dr. Avdesh Kumar. Dr. Avdesh Kumar is an additional director at the Directorate of National Vector-Borne Disease Control Program, Ministry of Health, Family Welfare, and he's looking after the Malaria Elimination Program in the country. He has more than 25 years of professional public health experience in program planning, management, standing system, and he is heading Malaria Elimination Program in the country, and his success story of the program are well recognized in World Malaria Report in 2018, 19, and 20. So over to you, Dr. Avdesh Kumar.
Good afternoon, everyone. I would like to thank at the outset ICMR for organizing this symposium, and also for inviting me to be actually part of this. Now, what I have been given the task is to speak about the malaria elimination program, the status, as well as the progress what we have made so far. I would like to share some of the slides. So let me just share my screen. The next one, please. Now, actually, we were in control mode for malaria, and the National Malaria Control Program, earlier as it was known as, started in 1953. And if we look at in free India or independent India, this was the first national health program of the country. after the launch of the Global Technical Strategy of WHO, that was in 2015, the country launched National Framework for Malaria Elimination in February 2016, and that's when we moved from control mode to elimination mode. And in next year, 2017, we launched the NSP, National Strategic Plan, which is a 5-year document up to 22. And next year, we would be coming up with the next 5-year document. The vision of this NFME is to eliminate malaria nationally and contribute to improved health, quality of life, and elevation of poverty. The goal is, as the name also implies, to eliminate malaria. That means the 0 indigenous cases throughout the country. And that the target is by 2030. And thereafter to maintain malaria-free status in the country. Now, since the country is huge, we have 36 states, and it's a difficult and different type of terrain. So we divided all the 36 states of the country into 3 categories of state. So category 1 states, numbering 15, which were low transmission states. There we targeted malaria elimination by 2020, that is last year. And category 2, numbering 11 moderate transmission states, we were targeting by 2022. And then the remaining some hardcore states, which are high malaria endemic states, we targeted by 2027. And the remaining 3 years we have kept for maintaining the 0 status and then to get the certification from WHO, etc., etc. Next, please. Next one, please. Now, if we look at the map of the country, these different, actually 3 colors. The green color is category 1, the pink is category 2, and the red is category 3. The name of these states are here on the screen. I'm not going to read this. And this already I have explained that category 1, which we were targeting by 2020, category 2 by 22, and category 3 by 27. Next, please. Next one, please. Piyush. Now, as per the NSP 17 to 22, the basic difference between the National Framework Malaria Elimination and NSP, the National Framework was basically based on the states broadly. The NSP, we came down to the districts, and all the districts of the country and the operational units, they were stratified into 4 categories. And the goal was to eliminate malaria in category 1 districts. That means those districts which were API less than 1 at that point of time by 2020. Category 2 districts by 22, and category 3 districts. The target was to stabilize the API at less than 1 by 22. At that point of time, this was the distribution of the districts in category 0, category 1, 2, and 3. So 75 districts were those districts which were historically, these are basically high hills, etc., etc., where the malaria was never known to occur. So 448 districts were in category 1, 46 districts were in category 2, and 109 districts were in category 3. I've also listed down below the states where these hardcore or category 3 districts were located. These states are Odisha, Chhattisgarh, Jharkhand, Madhya Pradesh, and some northeastern states, etc. Next one, please. Now, what was the strategy, or what is the strategy for malaria elimination? This is early diagnosis and complete treatment, and then case-based surveillance and rapid response. integrated vector management, which consists of indoor residual spray, long-lasting insecticidal nets, and larval source management. Then epidemic readiness and early response, monitoring, supervision, and evaluation at all the levels, advocacy, coordination, and partnerships with all these stakeholders, and behavior change communication and community mobilization is one of the most important strategies. And then program planning and management. So based on these broader strategies, we started moving towards malaria elimination. Of course, there are finer strategies depending on the stratification of the state as well as the districts. Next one, please. Now, after the launch of national framework, we augmented the supplies, we augmented the human resource, we augmented the other strategies and interventions. And this slide gives a glimpse of the major interventions made. Since then, 97 million lines have been distributed in the country. And all the subcenters which are more than one API, they have been saturated. That means all the households and all the members of those households, they have been given lines. We also gave the vehicles for the movement into the field, motorbikes, microscopes, additional human resource, etc., etc. Next one. These are some of the pictures of line distribution and usage at the community level. As I said earlier, 97 million lines were distributed in 7 northeastern states: Odisha, Jharkhand, Chhattisgarh, Madhya Pradesh, and 11 more states. So that means total 22 states these lines were distributed. Next. The ICBCC activities for community mobilization is one of the most important or crucial strategy. So through different channels and through different medium, this ICBCC component is being met so as to mobilize the community, so as to involve the community, and so as to motivate the community to participate in the malaria elimination activities. Next one. Next one, please. Now, if we look at the malaria graph in terms of total cases, the Falciparum cases, Vivax cases, and the deaths, from 2000 up to 2020, the malaria has declined overall. Now, here it was in 2016 on the right hand side of the graph, where or when the NFME was launched. Now, since then, up to 2020, we have achieved a decline in malaria cases by 84.5%. And similarly, in terms of deaths, it is 83.6%. Next one. If we look at the malaria map of the country, on the left-hand side is the 2015, which is the baseline before the launch of the framework elimination. And this is the 2019 map. I think we can make out that the red spots have almost gone, except now they are localized in few pockets. Down below, I have given the number of states which were in 2015 and 19, and similarly the number of districts in those 2 years. So less than 1 API states, which were 28, have now become 33. In fact, this number has now in 2020 gone up to 34. Now coming to the districts, in 2015, 77% of the districts were less than 1 API. But now in 2020, if I say, in 2019, it was 94%. Now, if we take it more than 1 API districts, these were 42 districts in 2019. Now, this number has further reduced. Now we have 30 districts in 2020, which are more than 1 API. All the remaining districts have less than 1 API now. Next one, please. Now, this category 1, category 2, and category 3, the malaria decline is across all the categories of states. So I'll not go into the details specifically because the malaria cases as well as malaria deaths, they have declined in all the categories of the states, category 1, 2, and 3. Next one. This is category 2. Next one. This is category 3 states collectively. Next. Now these are the districts now which are more than 1 API. This is 2019 data, but as I said, 2020 data is only 30 districts. So these are the 42 districts which are located in 13 states now, whose API is more than 1. I've also given the API range of these districts on the right hand side. Next one, please. Now, since the category 1 states were targeted for 2020, the question would be that what happened? Although we could not stick to that target of 2020, but there was remarkably significant decline in all the 15 states. I've just given a glimpse in this slide here that in 2020, this is up to August, there were 4 states which had just less than 10 cases. There were 6 states which had number of cases between 11 and 30. And there were 4 states which were between 31 and 150 cases, and only 2 states which were having more than 150 cases. So although it was not zero malaria, but yes, it was very near to zero malaria. So that was the status last year mid. Next. Next, please. Now, often this question is asked at various forums that how did India do it? I think this slide summarizes the success story of this malaria decline in the country and moving ahead towards the malaria elimination. After the launch of National Framework Malaria Elimination in 2016, we started this experiment from the 7 northeastern states, which is at number 1. I think this can be made out. So it was here at the middle of the graph, which is 2015. And after launch of Framework Malaria Elimination, there was sharp decline of malaria cases. In terms of quantification, 89% decline in malaria cases, 92% decline in deaths. I'm comparing the figures of 2015 versus 2020. Then we moved on to Odisha, which at that point was contributing 42% of the malaria caseload of the country. We obtained similar decline, so that's 90% in cases, 89% in deaths. Then we moved on to Jharkhand, where we again observed 86% decline in cases. Then we moved on to Chhattisgarh, where we could achieve a decline of 75% in malaria cases. Then we moved on to Madhya Pradesh next, where we achieved 93% of decline in malaria cases, and the deaths became 0. There are other states. So this what I have told is actually 11 states in total. So remaining 25 states of the country also, this was the case. Now these all 11 states were under the GFATM support. So the decline of malaria was much more as compared to other states because these were all under the intensified malaria elimination project. Next one, please. Now this just I am showing a slide that because this GFATM supported project which ended last month, 31st March, and the new project which has started from 2021st April. So what we have done. If we look at the table on the left hand side, these 10 states, 7 northeastern states, Odisha, Chhattisgarh and Jharkhand, which have been included under the new GFATM supported project. They are contributing just actually 12% of the population, then 48% of the total malaria cases, 81% of the Falciparum cases, and 77% of the malaria deaths. So these are the states which are being focused under the GFATM support, and this project continues for next 3 years. On the right hand side in the map of the country, this is where 2 circles where these states are located. So what we are going to do is we are going to cover 10 states, 155 districts, and roughly 164 million population under that project. Next one. Now this malaria decline in India has been well recognized by the World Malaria Reports of 2018, 19, and 2020. I'll not repeat these things. I hope everyone has read or gone through these World Malaria Reports of WHO, which have recognized India malaria program's success story. In fact, India is amongst the high burden countries if we look at the only success story. Otherwise, most of the countries, the malaria cases, either they are stabilized or they are going up. Next one, please. Now, what has been the impact of this journey towards malaria elimination at a glance? The malaria cases have declined by 84.5%. The malaria deaths have declined by 83.6%. These are the figures comparing 2015 versus 2020. Now there are 34 states which are less than 1 API in the country. So that means 2 states which are more than 1 API. In terms of districts, 116 districts have reported 0 case in 2020. So that's a huge number. Only 30 districts remain which are reporting more than 1 API. Now we are going ahead for the sub-national malaria elimination certification. And for this, we have instituted a cash award for the districts as well as for the states. This would be given to those states and districts which report 0 indigenous case during the first year, as well as to those who maintain the 0 status for 3 years. So this is the next which is coming up now. The guidelines have been readied. The communication has gone to states as well as to the districts. We are requesting them to now submit their applications, and then there would be actually verification by the independent teams of expert, and then the certification process would start. Next. But no doubt the journey is impressive and seems to be successful globally. There are few key challenges. And the biggest and most important challenge is how to reduce or how to decline the remaining 15% malaria in the country. It also gives us a lesson, and the biggest lesson which we have learned is that we can do it. And I repeat, yes, we can do it, which I think few years back was looking a very difficult task. Coming to the challenges, the biggest challenge now seems to be the complacency. With this much of success in all the states, all the districts, the health administrations as well as the disease control programs, the priority of malaria in the list has gone down, and that happens. I think one of the speakers was referring to 1965 in the fight against malaria, when we had achieved 0 deaths. So we need to take care of that complacency factor. Besides that, for another 10 years at least, we would have to sustain adequate funding for achieving malaria elimination. Availability of trained human resource at different levels, starting from the block to the district, state, and also to the national level, is a challenge. So we have done, or we have taken care of this aspect in the Global Fund supported project. Then there are large conflict affected, hard to reach, difficult, heavily forested areas. I think Dr. Amit Sharma was referring that in his presentation, where the health infrastructure is not that good. So that challenge is also there. Another is the private sector. Although now 31 states have made malaria a notifiable disease, the mapping of the private sector, all reporting units have been done. And we are actually trying to get monthly reports regularly from all these reporting units of the private sector. But still, that coordination needs to be improved. The drug resistance, especially the artemisinin resistance in the neighboring countries, I think one of the previous speakers were referring to the Myanmar and northeastern states. Myanmar has now reported this resistance. So there we need to check on this. Insecticide resistance is another challenge which needs to be tackled. Entomological infrastructure in the country is not that good, I would say. Again, we are taking care of this also under the GFATM supported project. This would be required for surveillance and response mechanism. This would help us both ways. Number one, to achieve malaria elimination as per the timeline. And also, once we achieve malaria elimination, how do we sustain malaria elimination? Then entomological infrastructure would be very important. And additional benefit would be that the other vector-borne diseases would also be taken care of by this entomological infrastructure. Migration of the people from district to district, state to state, or even between the countries. That aspects are very important for malaria, or what we call them is the cross-border collaboration. I think Bhutan was referred to earlier. We are supporting Bhutan so as to achieve malaria elimination as fast as they can. And we have now at the district level, I think WhatsApp groups have been made so as to share on ready basis the information. The climate change is a larger challenge. Now in India also, in some of the higher reaches of the districts, we have observed that mosquito, which was not earlier there, now the mosquito has reached. So that challenge remains and we need to tackle it. COVID-19, as we all know, since last year, now it's almost 15 months, has been a big challenge because there was no specific national health program for the COVID-19. So the diversion of human resource, especially the frontline health workers, was made. And there was delay in the indoor residual spray, as well as the LLN distribution, monitoring, and supervision activities. Now, since this is ICMR supported symposium, I think where are the technical challenges? I would list some of them. Asymptomatic malaria. In some of the areas, we still have asymptomatic malaria. We are taking care of that by the mass screening and the treatment strategy. RDT versus the blood microscopy or blood slide or some other very highly sensitive and specific diagnostic method, which can be applied at the field level. Cross-border malaria, I already spoke about in brief. Drug resistance, insecticide resistance, already covered. Another is the Vivax malaria. As we move down towards the malaria elimination, the Falciparum declines fast. The Vivax remains. So that challenge is there. And of course, now we have the fifth species of the Plasmodium knowlesi, which needs to be tackled. So those are the broader technical challenges which need to be looked into so that this actually timeline is not derailed as far as malaria elimination goes. Next one, please. I think the question would be that what was the COVID impact on malaria surveillance? The answer is that yes, there was some decline in malaria surveillance because of the COVID. But the decline in 2020 compared to 2019 was much more than the decline in malaria surveillance. So that actually data we have analyzed, and we have found that there might have been some impact because of the decline in surveillance, but otherwise the malaria decline is also real in 2020. Next. Some of the best practices during COVID-19, actually, which gives us an idea that how we were maintaining the services almost to the fullest level. Mizoram distributed 4.14 lakh LLIs within 5 weeks in the community, and they followed all COVID-19 actually protocol. Similarly, Tripura distributed 8.19 lakh LLIs within 8 weeks, again following the COVID-19 protocols. And then Malaria Mukt Bastar Abhiyan in Chhattisgarh, which is basically the mass screening and treatment strategy, was successfully undertaken in the Bastar region. Bastar region of Chhattisgarh, consisting of 7 districts, is the area which is chiefly contributing malaria in Chhattisgarh. Next, please. Next, please. Now that was, I think, few slides which I thought may be useful for this symposium. I'm sure there would be many more issues which the audience would like to ask. And if there are any questions, I would be glad to answer that. So thank you very much. Thanks a lot.
Thank you very much, Dr. Avdesh Kumar, for a wonderful presentation and describing various components and progress of the National Malaria Program. Thank you for being here. Now, because we are already late, I request another speaker here. This is Dr. Marta Bahia. Marta?
Hi, 1 second.
Yeah. So Dr. Marta is a research scientist based at Kemri Wellcome Trust Research Program in Kenya. She is a member of Cochrane Group and has contributed to evidence synthesis on the use of repellents for malaria prevention. Currently, she is leading the entomology work package of a CARCT evaluating ivermectin mass drug administration for malaria control in Tanzania. And the title of her talk is Mosquito Repellents for Malaria Prevention. Over to you, Marta.
Thank you so much for the introduction. So today I'm going to talk about mosquito repellents and a systematic review that was conducted to see their effect on malaria prevention. The review was done for the Cochrane Collaboration. It was a Cochrane review, and I'm sure a lot of you are familiar with these reviews. Cochrane reviews, they sit at the top of the pyramid of evidence, and they summarize primary research in human healthcare and policy, and are recognized as the best and the highest standard for reviews. The Cochrane Collaboration is a non-profit, non-governmental organization. It's formed to organize medical research findings and to facilitate evidence-based choices about health interventions. It was formed in 1993 in response to Archie Cochrane's call for up-to-date systematic reviews on randomized controlled trials, and mostly to support the British NHS guideline decision makers. The pyramid of evidence starts with systematic reviews, and just after systematic reviews, we have randomized controlled trials, which is which are considered the highest quality of evidence trials, followed by observational studies, cohort studies, case control studies, case series. At the bottom, we have expert opinion, which many think is very informative, but actually they lay at the bottom of the pyramid. And nowadays, unfortunately, we also have Twitter and Facebook as sources of information, which should be at the very bottom of the pyramid of evidence. So our talk today is on mosquito repellents, and I'm sure a lot of you know what this is and probably use them at home. Maybe you are even fierce advocates of them. Mosquito repellents come in a lot of different forms. They can be topical repellents that you apply on your skin. They can be spatial repellents, like these coils that are burned, and spatial repellent compounds are dispersed into the environment and repel the mosquitoes. They can be also electric, where they are placed in indoors in a socket and emanate. They can be passive emanators, and nowadays we have a bit of a boom in accessories for mosquito repellents, including these mosquito repellent bracelets and even shoes. The repellent market is valued at around $3 billion US, and it's projected to reach 5 billion next year. So it's quite an important market, and there's a lot of interest from the industry to move forward with repellents for malaria prevention, as it would be, although it would be another commodity. We all know, because you also use them, that there's a lot of entomological evidence showing that mosquito repellents can reduce mosquito bites. It's an intervention that has overwhelming evidence showing that mosquito bites can be prevented by using repellents. And these studies have been done in a variety of locations, on a variety of vectors, and using different compounds. So the interest in mosquito repellents for malaria is quite simple. We heard today about residual malaria transmission. So mosquito repellents, they may complement the traditional interventions, because they will be an intervention that will target the outdoor biting mosquitoes, and that might address the gap that current interventions preclude. In addition, mosquitoes are changing their host-seeking and resting behaviors, and so in response to their selective pressure to LLINs and IRS, and so mosquito repellents would be another option for people in these regions. So we defined our systematic review by first defining our PICO. PICO stands for Participant, Intervention, Control, and Outcome. Our PICO our population was children and adults living in malaria endemic areas. We excluded travelers. Our intervention was topical repellents, insecticide-treated clothing, or spatial repellents. Then our control were either placebo or no treatment, and we looked at epidemiological primary outcomes, malaria case incidents caused by Falciparum or Vivax, and malaria infection incidents caused by Falciparum or Vivax. We also looked at secondary outcomes, including all-cause fever, anemia, and adverse events, but I won't have time to talk about these today. In the review, we included all available randomized controlled trials and cluster randomized controlled trials. The population needed to be permanent or temporary residents of malaria endemic areas, but not travelers. So, if, for example, they were stationed there as military, they would be considered temporary residents. In terms of interventions, for topical repellents, we included any formulation, including DEET, 15% or higher, PMD, icaridine, or picaridine. For insecticide-treated clothing, we only included permethrin-treated clothing. And for spatial repellents, we included any spatial repellent formulation, from coils to emanators, including transfluthrin, metafluthrin, dialethrin, or pyrethrin. And we made sure that all interventions had ITNs in both arms, so that would be balanced. Our methodology, well, we started off by importing all of the search results into our Covet software, and then 2 independent authors did a title and abstract screening, following by this inclusion decision after full text screening, and then the data was extracted for each study. We also listed all of the ongoing studies. We excluded some studies, and we gave reasons for that. Reasons could have been that the trial was not an RCT, that it had, if it was a cluster randomized controlled trial, if it only had 2 clusters, then we would also exclude, or they did not define the intervention of interest. All of the data was extracted into pre-piloted forms and by 2 independent authors, and with that data, we then looked at we assessed the risk of bias using the Cochrane tool. We looked at selection bias, for example, method of randomization. We looked at performance bias, for example, are the participants blinded? We looked at detection bias, is the person reading the outcome biased? Do they know? Are they blinded? Attrition bias, we looked at loss to follow-up in the different arms. Reporting bias, we did funnel plots to look if there any evidence of reporting bias, and we also looked at other bias such as baseline imbalances or so. Then we did a meta-analysis with the data extracted. The meta-analysis was it was done by intervention type, so we did different meta-analysis for each outcome and intervention type. We subgrouped within each meta-analysis if the studies had included LLANs in both arms or not. And we calculated risk ratios and presented these in forms of forest plots. We then combined the data from cluster randomized controlled trials and randomized controlled trials after adjusting for clustering by calculating their effective sample size. And we looked at heterogeneity, how different were the studies from each other. And this was also assessed in a meta-analysis using chi-square and I-square statistics. We assessed the quality of evidence using grade approach. The grade approach looks, well, it starts off as 4 points for any review that only included randomized controlled trials, like we did. And then from these 4 points, you can downgrade or you can upgrade. Well, you can't upgrade above 4, but so reasons for downgrading is if the study design, the quality of the study design is not high. If you're not including randomized controlled trials, you downgrade to 2. But in our case, we started off with randomized controlled trials, so we start off at 4. We can downgrade if there's a high risk of bias. We can downgrade if there's a lot of imprecision. This happens when we have very low sample sizes or large confidence intervals, and our measure of heterogeneity is very helpful to decide on downgrade. We also downgrade if there's inconsistency. This happens when there is a lot of heterogeneity between studies that are are similar. And we also downgrade for indirectness if the question does not directly answer if the trial does not directly address the review question. And reasons for upgrading, they rarely occur, but if we see a very high magnitude of effect or dose gradient response, then you can upgrade. So from our results, we found 440 records. We screened, we found another 3 from other sources. We screened the 443, removed the duplicates, and then excluded 6 studies with reasons, included 10 studies into our meta-analysis. From these 10 studies, 6 were on topical repellents, 2 were on insecticide-treated clothing, and 2 were on spatial repellents. We then summarized our risk of bias, and from all of the studies that we had, we denoted that there was quite a substantial risk of bias for selection bias, so the random sequence generation that was used, and also the allocation concealment method, and that was in the end a reason to downgrade. From a meta-analysis, I'll start off with topical repellents. Meta-analysis for clinical malaria caused by Plasmodium falciparum. In a subgroup analysis was separated by LLIN usage. Only one study did not include LLINs, that's the Mark Rowland study, and it reported a 60% protective effect against Plasmodium falciparum malaria. However, studies that did include LLINs, we didn't see such an effect. Both arms had confidence intervals around the point of, both studies had confidence intervals around the point of no effect. In regard to clinical malaria caused by Plasmodium vivax, we saw a very strange result. Paradoxically, repellents increased the risk of Plasmodium falciparum malaria, Plasmodium vivax malaria. However, it's unlikely that this finding has any epidemiological importance. It's more likely that it was just a product of improper study methods, because none of the studies cleared existing P. vivax infections at start, and it's unclear if recrudescent infections were imbalanced in the both study arms. And then in regard to malaria infection incidence, no protection was seen. Studies were very imprecise, and they had very large confidence intervals, except for this one study from McCready done on pregnant women, which also did not include LLINs. However, the estimate crosses the point of no effect of 1. Then, in terms of malaria infection incidence caused by P. vivax, there's also no protection seen, and some studies had very wide confidence intervals, and again, it's crossing the point of no effect. So summarizing the findings in regard to topical repellents, we have very low quality of evidence. And that means that we have very little confidence in the effect estimate. The true effect is likely to be substantially different from the estimate of effect. We have low confidence of evidence in regard to malaria infection incidence, and very low in regards to clinical malaria caused by falciparum. Then I think one of the points that we need to discuss in this review, and it's one of the important things that came up, is the issue of compliance and adherence to the intervention. Self-reported, most studies used self-reported compliance, and they found it to be high. And they used methods such as interviews and questionnaires, and these were backed up sometimes by other indirect methods like weighing bottles or counting the bottles, the bottles of repellents, that is. And these methods, they are quite unreliable, because participants can well, they can lie to please the investigator, which is what we call a response bias, saying that yes, they did apply it, they did adhere, although they didn't. Or they can dispose of the repellent, or they can share the repellent, and also there's a lack of standardization because some people may apply double the amount than others. So basically, it's quite an unreliable nature. It's quite the data has quite an unreliable nature, and it might overestimate the reported compliance. There was one study conducted by Sluitz in 2016. Where that was the only study where this occurred. They sent somebody to observe compliance. What we somehow a spy. It was sent to the village to record observed compliance, and he showed that actually, despite more than 70% self-reported, only about 6 to 15 of the participants were complying with the intervention. They would say they were very pleased with the repellents, but reasons for not applying them were usually that they had either forgotten to apply or just didn't do it appropriately. And then in an intervention like topical repellents, the poor compliance will lead to a decrease in study power, and that requires studies with a very large number of participants, which is practically unfeasible, and especially as malaria prevalence is reducing. We should also note that Sluitz had 30,000 participants in each arm. We actually digged a little bit into sample sizes, and we looked at the malaria prevalence from each study site to control in each study's control arm, calculate the necessary sample size, and noted that all studies were severely underpowered, even the very large trial that was conducted in Cambodia. That trial effectively would have needed around half a million participants to reach its objectives. And these reasons included the very low prevalence of malaria in the study area. It was less than 2%, and also a flawed study design with very large cluster sizes, which reduced the effective sample size after adjusting for clusters. These sample size estimations are assuming 100% compliance, which in reality is also unrealistic, making it arguable if RCTs or if randomized control trials can actually be used to test topical repellents. Moving on to insecticide-treated clothing, we then analyzed, did a meta-analysis of insecticide-treated clothing on Falciparum and Vivax. We noted that there was a protective effect of about 50% against clinical Falciparum malaria. It's important to note that neither of these studies included LLINs, and they were both done with displaced populations. I believe one was with refugees and the other one was with military. So it's unlikely that the exposure, it is likely that the exposure and the living conditions that these participants were in were considerably harsher than usual. And this made us downgrade that quality of evidence for indirectness, because it wouldn't apply to all of the population. The protective effect against Vivax was not as large as Falciparum, and the confidence intervals crossed the point of no effect. And it's also important to note that similarly to these studies on topical repellents, neither study cleared Vivax infections at start. Summary of findings: we downgraded for risk of bias and also for baseline bias, because there was not reported how long soldiers were in each arm were deployed, and our data basically is of low quality, and so that means that our confidence in the effect estimate is limited. The true effect may be substantially different from the estimate of the effect that was here reported.
May I request,
Yeah.
May I request you to summarize, please, quickly?
Sure, I'm nearly finished. Then we looked at spatial repellents. We had 2 studies, both with very large confidence intervals and quite discordant results. An overall risk ratio ranging from 0 to nearly 2, and that denoted a high degree of inconsistency and imprecision. We also downgraded for risk of bias, and because Hill 2014 was not blinded, and for imprecision because of the large confidence intervals, and inconsistency because of the high degree of heterogeneity between the 2 studies. So in conclusions, there are insufficient well-designed studies on topical repellents to draw evidence-based conclusions and make well-informed recommendations to policymakers in regard to their use of the malaria prevention tool. We also need to consider new methodologies other than randomized controlled trials or cluster randomized controlled trials if we are to evaluate the use of repellents for malaria prevention. And it's important to note that low compliance alongside decreasing malaria prevalence levels in potential study sites are major limitations for the design of randomized controlled trials or cluster randomized controlled trials, because an unfeasible number of participants would need to be followed up to reach statistical powers. In regard to insecticide-treated clothing, there's lean evidence that the use of ITC is useful in displaced populations such as or disaster situations, that they may provide some degree of protection from malaria in the absence of LLINs, but we need stronger evidence to support this. Furthermore, the results only indirectly answered the review question because the participant groups were not the general population. And in regard to spatial repellents, well, we have very low quality of evidence, and we cannot draw any conclusions on the data that is now available because it is the studies were not designed appropriately. So to conclude, I leave you with a question. We have seen that we have interventions that show high efficacy, providing personal protection. We know that they can prevent mosquito bites. We have seen this, which is measured. But at programmatic level, they have very low effectiveness. So in a real world application, any intervention, we need to know, does it work? We know that it works. Is it ethical? There's evidence that there might be diversion of vectors, and this is also an important question because of the lack of adherence or compliance. Is it appropriate? Can everybody access repellents, and can everybody afford repellents? And is it accepted? Will people actually comply and use it? I can see that the application of repellents can be might be of interest to displaced populations or perhaps to particular high-risk populations, such as forest goers, but I don't think that we will be able to answer that question with a review that reviews the evidence on a programmatic level. So additional well-designed studies and well-designed reviews need to improve the quality of evidence in order to allow decision makers to rely on recommendations. Okay, thank you.
Thank you, Marta, indeed, for a nice presentation. And thank you for dwelling into repellents as the personal protective measures and evidence generated through the Cochrane review. In fact, repellents have been marketed as a supplementary measure, not only for malaria, but against dengue too. So with this, I would like to invite the next speaker, Professor Ashish Das. Ashish, can you join? Yeah. So Professor Ashish Das is a professor in BITS Pilani campus, and his interest includes different aspects of research into the human malaria parasites involving clinical cases, transcriptomics, and related approaches. So he'll give us a taste of a mixed clinical and basic research. So I request Ashish to Please go ahead.
Just a minute, I have a slide problem. Display, yeah. Am I visible? Is the screen visible?
Absolutely.
At the onset, let me thank you for your kind invitation to be in this panel of very distinguished panelists. I thank Dr. Amit Sharma, Director in IMR, and Dr. Sachin Sharma, who's been the lead spirit behind this event. What I will be talking about is part of the work which we have been doing or attempting to do, and our limited efforts in trying to understand some aspects of severe malaria. However, today's talk will not focus on the severe aspect, but will focus on potential new molecules which we could have clues to, or we feel we have clues to, as the result of transcriptomic-based networks. But before I go into that, let's look at the issues which all of us are aware of, and many speakers have elaborated on a number of these themes. Vaccines, drugs, diagnostics, drug resistance, need for new targets, need for ultrasensitive detection. All these remain burning issues in spite of the fact that there has been significant efforts and some successes in reducing the malaria burden, especially in places. Now, the fact remains that the nuclear genome of Plasmodium falciparum and Plasmodium vivax encodes about 5,300 genes. And out of these, about 50 to 60% have no known functions. What that means is there's a potential for harnessing a lot of unknown molecules for the benefit of society. What I'll be talking about will be based on samples collected from patients and further studies done with them, primarily transcriptomic studies using microarrays. Now, which means essentially that we are primarily focusing on the asexual blood stages, and in some cases, gametocytes, especially the early gametocyte stages, which are difficult to distinguish under microscopy. Our efforts, which have been going on for over a decade now, started in the end of the previous decade when we really wanted to focus on severe malaria being caused by Plasmodium vivax, which we had reported in various fora. Now, severe malaria by Plasmodium vivax to us showed many similarities to that being caused by Falciparum malaria, with some interesting differences. As I said, our study starts with patients and their voluntary consent to allow us to have access to their blood material so that we can process it for the parasite RNA. In this slide, we have briefly described our baseline for collection of these samples, which include diagnosis through peripheral blood film, RDTs, and finally polymerase chain reaction. Until and unless all of these were concurrent, we did not allow any samples into the study, as well as various laboratory investigations. This shows some of the RDTs, the patient microscopic slides, and one of our PCR-based evaluation. This is a mixed infection, which consists of Falciparum and Vivax in the same sample. Now, what we did was we designed some cross-strain P. falciparum arrays. This design was done with our collaborators and outsourced providers, Genotypic, And the samples were mostly, in fact, all of these samples which I'm talking about was from a tertiary care hospital, which was where our collaborators are located in Bikaner in Rajasthan. The samples which I will be talking about were collected in the period 2009 to 2010, approximately. And without going into too much of details on the cross-strain P. falciparum array, I would just like to point out this is the only array which has transcript sequences for 3D7, 8B3, and parts for the ID4. And this is another microarray which we developed for the Plasmodium Vivax, which has 4180P Vivax Sal1 genes in sense orientation. I will right now ignore the anti-sense orientation genes. Now, what we did with this, because our primary goal was to investigate the phenomenon of severe Vivax malaria, and we obviously got onboarded with P. falciparum malaria. Why were we interested in looking at patient samples is obvious, because the patients provide a milieu which is lacking in culture, which has other advantages, but which does not allow the host factors which can perturb various gene expressions. This schemata shows us what we did with some of the arrays. And what we did was we made gene expression networks. And these gene expression networks we further analyzed to reach certain important points. So in the beginning, let me go to I will not go into the steps for constructing a co-expression network. However, what we did was we utilized signed weighted networks. This was the total number of samples which was used for creating this network, which means microarrays covering the whole genome expression profiles of 21 samples, some uncomplicated and others showing complicated manifestations, were taken and used for creating these networks. This shows the hierarchical clustering dendrogram of genes in the network, which partitioned into distinct modules. And we had a total number of 27 modules which were created. This represents the IGEN gene network, representing the relationships between the modules, the IGEN gene being the hypothetical representative gene of a module. And then to get into data proper. We had, for the falciparum studies which I'm presenting here, uncomplicated patients with jaundice and renal failure, and patients with jaundice. When we compared the differentially expressed gene pattern between these two groups, and this comparison was done vis-a-vis the uncomplicated parasite expression from different places, averaging it out, we found that in the jaundice renal failure differentially expressed gene set, there were 227 genes which were differentially expressed, whereas in the jaundice differentially expressed gene set, there were 178 genes which were uniquely differentially expressed. And very interestingly, there were 153 genes which were differentially expressed and overlap between these two data sets. So we tried to compare these modular gene expressions. By this, we had made these different modules of co-expression networks. And we wanted to see whether the genes which partitioned into these modules were primarily upregulated or downregulated in severe disease. Now, at this point, it's obvious that if it's upregulated in severe disease, it means that it could give us clues as to molecules which are involved in the severe disease from the parasite side. And if it's downregulated in severe disease, which means upregulated in uncomplicated malaria, it could give us clues to molecules which are upregulated in uncomplicated disease. So both these analyses have their own inherent strengths. In addition, there are also steady state molecules between the two data sets. So when we tried to look at the geoenrichment terms for some of these modules, we saw that they refined to biological processes. There were ubiquitin-independent protein catabolic process, oxidation reduction, mitochondrial transport, intracellular organelle lumen, etc. But what we did then was we utilized the cytoscape tools to make these graphical representations and pin down on genes, which we refer to as hub genes, which are interconnected to many, many genes in these modules. So we have upregulated hub genes, we have stably expressed hub genes, and then of course we have the connecting genes. We wanted to see whether there was any biological significance to these hub genes. And we found in the very few which I'm showing here that some of them were in fact considered as potential drug targets, like hexokinase, aquaglyceroporin protein, etc., whereas others had significant uses. We analyzed these hub genes further, and we found that there were many conserved and hypothetical proteins which has come up as hub genes in our modules. Of special interest to us became the proteins, the plasmodial proteins, which are referred to as conserved plasmodial proteins with unknown function, which show very little similarity with human proteins. So there's very little similarity with the host proteins. And we felt that some of these, and we still feel strongly, that some of these could provide us clues to possible strategies, either diagnostic or therapeutic, of the future. The data which I just presented in very brief has been published in this paper some time back. And this shows some of the collaborators, the Professors Kochar from Bikaner. We went further, and this is unfortunately data which we haven't got down to publishing, though we intend to do it very soon. And this consists of the analysis of 26 samples, which includes 6 non-severe Vivax cases, and the rest either with cerebral malaria and related complications or with jaundice in some form. Similar to our analysis with the Falciparum cases, we tried to find out what is the result of a Venn diagram when we take the differentially expressed genes, which was the result of comparing the hepatic dysfunction and cerebral malaria cases individually with the average of the uncomplicated expression. And we found that we had 589 uniquely perturbed genes in hepatic dysfunction, 72 in CM, and an overlap of 60. We further divided this data into modules. And what we found was that modules like dark magenta and dark gray represented primarily upregulated genes, whereas orange, red, and blue represented primarily downregulated genes. Now, to make this point again, upregulation in the severe disease made us call refer to these modules as severe disease-specific modules, and downregulation in severe disease caused us to refer to these modules as non-severe disease-specific modules or uncomplicated disease modules. And I'll skip this slide. This shows again, a similar representation. So this shows the top 3 hub genes with their first interacting partners in the severe disease-specific modules, dark magenta and dark gray. And we see what is the potential of these hub genes based on the networks where they form the hub in. And when we look at the geontological terms, we find that the dark gray goes into chromosome, whereas dark magenta has a host of terms which it enriches to. The non-severe disease-specific module again shows multiple genes with different attributes given to them in clusters. Now, when we analyzed this at an individual level and looked at the top hub genes, we found that many of them indeed were, again, just as in the Falciparum network, hypothetical proteins conserved in Plasmodium. Okay? So we thought we would try to do some experiments, and this has just been recently published. And this is the result of experimentation with antipeptide antibodies to a conserved Plasmodium protein of unknown function, which was upregulated in both jaundice and jaundice with renal failure in the data set which I showed previously during the Falciparum differentially expressed gene setting. And what we found was, when we did quantitative real-time PCR with 3D7, we found with 3D7 material, we found that this was present in ring, trophozoite, and schizon, and to a much lower level, but present in gametocytes. This was at the level of the transcripts, which bore out our data, which we had seen from our microarray data. Subsequently, we used the antipeptide antibodies to localize the possible protein. And we found that this appeared to be a cytoplasmic protein and was present in both asexual as well as sexual stages of the parasites, albeit at different intensities. In fact, it was present in all the asexual stages as well as the sexual stages of the parasite. This, to us, is very, very interesting, because what it shows is, A, the transcript data can be directly related to protein levels, at least in some cases. B, a conserved hypothetical plasmodial protein with unknown function can have very, very important usage if further information about them were known. To conclude, I started my talk by saying that about 50 to 60% of the genes in the genome of the Falciparum and Vivax Plasmodia we don't have much information about them. Well, using these in vivo transcriptomic networks and focusing on the conserved plasmodial proteins, which have very low hits to humans, if at all, we feel that we could narrow this number down to maybe 35, 40, which I feel is a significant pointer to very important work, which could provide us leads to future diagnostics, markers, future therapeutic targets, and many other strategies which we can't envisage right now. I would like to conclude by acknowledging our collaborators, my colleagues, my former students. The network work was done primarily by Dr. Bhupati and Dr. Subudhi. Dr. Deepak, Dr. Isha Pandey, who was the primary driving force behind the immunolocalization work. My collaborators, Dr. Agam Singh, I'm just mentioning some of my collaborators right now, and my present team who are at BITS. And last but not the least, all the patients who volunteered for the study, the Department of Biotechnology for the financial support, and all scientists involved in data generation or deposition and curation in various databases, without which this sort of a study would not have seen the light of day. I thank you all, and I end my talk now.
Thank you, Professor Das, for a nice talk and sharing your beautiful data on gene co-expression and their network analysis. Thank you so much.
Thank you.
Yeah. So next is
What we do.
Professor Sanjeev.
Thank you very much, Sachin. Thank you for the invitation to speak to this symposium. I hope that we can get the slides up from your side.
Yush, can we?
Sure, sir.
And while we're waiting, I just wanted to also say what a pleasure it is to catch up with some old friends and to see some new faces also, particularly nice to see Amit. You know, hear of the successes of the work that's going on.
So before he start, I'll just briefly introduce Professor Sanjeev Krishna. Is an old friend and is a professor of molecular parasitology and medicine at St. George's at University of London. He is an infectious disease expert with a special interest in molecular to cellular studies on malaria. And in today's talk, he will shed light on how effectively combat the dual challenges of malaria alongside new complication posed by COVID-19. Over to you, Sanjeev.
Thank you for that intro also, Sachin. Yes, none of us can ignore the ravages of COVID-19. And actually, I was speaking yesterday to NDTV about the horrors of what's happening in Delhi at the moment, for example, that we're all seeing on the global screen. So, you know, these are very difficult times. This is a biological tsunami, and some of us in different countries have been through it a little bit earlier, maybe even more than once. And of course, as these pandemics come and go, we are nevertheless left with some recurrent problems and challenges that were there before the pandemic arose and will no doubt be there even when the pandemic gradually abates. We hope quickly abates. Now, just before I begin my talk, I should just say that I am involved with 1 or 2 diagnostics companies in my declaration of interests there. And also, I'm a member of the guidelines development group for the World Health Organization for the subject of anti-malarial chemotherapy. But that, of course, the views that I'm expressing today are very much my own and don't represent any of these organizations. Could I have the next slide, please? I thought I'd start off, next please, just by saying, just one at a time, just by saying that the talk initially is not going to be about COVID-19. I've alluded to the shocking increase. This is a slide that shows how rates of COVID-19 have increased in India. And of course, that increase is showing at the moment no signs of flattening off and may carry on for some time. But the talk today is not about COVID-19. Next, please. And neither is it about a very important and interesting subject, which is to repurpose antimalarials to help us to tackle COVID-19. If there is an interest in this subject, then we, amongst others, have reviewed the topic quite recently. You know, it's of interest because often antimalarials have been repurposed successfully, for example, to treat heart disease in the case of quinidine, or skin diseases in lupus for the case of hydroxychloroquine. But it does come back to one of the other subjects of the talks earlier given today, and that is that we do need good quality randomized controlled trial evidence to, or higher level evidence to support the use of antimalarials in this context. And of course, that evidence has been lacking, even though the original ideas were seized by some pretty high profile politicians. Next, please. So the subject, of course, is malaria, but malaria in the context of COVID-19. And when the pandemic was emerging, next please, we got together as interested parties from Africa, scientists outside of Africa, led by the Nobel laureate Tu Youyou, shown there. She got the prize, of course, for discovering artemisinin, and I call her the mother of artemisinin, which she was recognized for in 2015. So we put together some thoughts about how important it was for areas that had not yet seen the pandemic, how important it was for those countries to be prepared to deal with malaria in the context of the pandemic. Next slide, please. And we were informed from lessons gained from a previous pandemic not so long ago. Now, one of the themes that has come out with pandemics, and I think it applies even during this pandemic with COVID-19, one of the themes that's emerged is that collectively, globally, we are very poor at learning lessons from pandemics ourselves. So we went back to the Ebola virus disease outbreak, 2014, 2015, and we noted that the collateral damage, if you like, of having an Ebola virus pandemic in these three countries in sub-Saharan Africa was that malaria was not being diagnosed, malaria was not being treated because of this, and that this resulted in increased mortality. Now, these observations were made in sub-Saharan Africa, but they of course apply across the globe. And we've seen in the United Kingdom, for example, the increase in waiting lists, the missed diagnoses that emerge during the pandemic, for example, for cancer or other acute conditions that would otherwise have been diagnosed, and so on. And remember that our observations were being written in March 2020, and at that stage, there weren't many cases in Africa, but it was becoming clear that the chances were the pandemic would spread. Next, please. Now, what were the points of preparedness that we wanted to highlight? One of these was to maintain the distribution of insecticide-treated nets, bed nets. And in fact, one of our previous speakers has shown strikingly, Dr. Avdesh has shown strikingly how important it is to maintain the distribution of insecticide-treated nets. And he's also emphasized that this process was carried out in compliance with COVID avoidance measures. And that brings us to the second point, and that is that really part of this process would have been to anticipate the requirements for COVID avoidance interventions. There are thoughts about where it may be necessary to put into place mass drug administrations as antimalarials if other measures particularly become strained. And the need to maintain the key malaria diagnostics, which so often are helpful when there are similar clinical features presenting for malaria or for conditions like COVID-19. And of course, to make robust any infection management protocols that include, for example, diagnostics in their pathways. Next slide, please. Now, if you are interested in hearing about some of the perspectives from different countries and how they've coped, particularly, for example, where the greatest burdens of malaria are in sub-Saharan Africa, then I would just point you to some case studies described by those who are involved at the front line in these countries in the URL, which is shown right at the top of the slide there on the WHO website. And indeed, it's the WHO recently released World Malaria Report that I've referred to summarize some of the data that I came across from Southeast Asia. Next section, please. And which really showed that one of the largest burdens of malaria has been in India. Next, please. We got some, I'm very pleased also to hear the updated figures, because it's nice to say that the figures shown on this slide, 2015-19, showing such large decreases in malaria and in deaths, are both out of date and illustrated in the previous speaker's talk with far more detailed data in region by region distributions for malaria. So it's been a remarkable challenge to keep this downward pressure on malaria cases and deaths, but India somehow seems to have really succeeded in maintaining this in the last few months. Despite, as we know now, the COVID-19 pandemic spreading to all regions and now becoming really a catastrophe in some particular areas. And it's the same challenges that were listed in our preparedness article that would be likely to impact on malaria control in the next few weeks and months. And these would include the disruption of supplies to control malaria, whether it's diagnostics or drugs, protection of healthcare workers, and when resources are tight, and they particularly are in India, of course, then there is a danger that these would be redirected away from these treatable deaths caused by malaria or illness. Next slide, please. Ayush, next, please. What I've shown here in the next slide is that it's very important for all malaria endemic countries in the context of the challenge of a COVID-19 pandemic, to maintain a very high level political, that's to say, to keep the decision makers, the politicians, the ones who are directing resources, also focused on malaria as an issue, not just on the emergency of COVID-19, and to maintain funding support for that as well, and to have dialogues with the donors who are underpinning some of these research, operational efforts in different countries to control malaria. Next line, please. And this, of course, emphasizes again, and we've not been particularly good, certainly at the start of the pandemic in the UK as well, in ensuring the safety of healthcare workers as they go about their business, either in the community or in central facilities. Next, please. I think diagnostics play a key role because, as I've already emphasized, of the risk of confounding and of undertreating either one or both conditions. And some countries have taken active steps to integrate care pathways that include malaria, perhaps other infectious diseases. Dengue was mentioned in one of the talks earlier. Dengue, for example, or other mosquito-borne infections, to integrate care pathways with the management of COVID-19 as well. And throughout this process of implementing management for malaria in COVID-19, we also know that it is the most deprived and the most displaced individuals that are likely to suffer the most. And we must maintain our focus and our support for those communities as well as for the population at large. Now, this is just another way on this slide of showing how cases have come down. Next, please. But what surprised me from this World Malaria Report summary was how much malaria funding there is in India, right there at the bottom, compared to other countries when it's normalized to per person at risk. This is a couple of years ago. So if you were to look at the achievements in India, I think they would be even more remarkable when you compare the resources that are available on a per capita basis for those at risk of malaria. Next, please. Now, I'm not going to spend too long in the next few slides. These will be just pointers really to what could be of interest in terms of the science of malaria in the time of COVID. And next, please, here's some colleagues of mine who've set out some interesting examples of scientific and technical questions that arise on the interface between malaria and COVID-19. And here, there's some details. SARS-CoV-2, the virus that causes COVID-19, enters lung alveolar cells via ACE2 receptors, not exclusively, of course. There are other receptors in other cells, but for alveolar cells, it's ACE2. And ACE gene variations include insertions or deletions, I or D alleles. And it's already known that the D allele leads to higher ACE activity and angiotensin II levels in individuals that have that particular allele. But it was established in India more than a decade ago that these alleles, the ACE-ID and the ACE-DD alleles, protect against cerebral malaria in an Indian cohort. So there are questions that could be addressed at a pathophysiological level that have arisen from this existing information that relates one infection, malaria, to the newer pandemic infection that is spreading in some of these areas. Next slide, please. Take another example, G6PD. is very well established as protecting against malaria. It was one of the earlier gene polymorphisms that was associated with protection against malaria. But we also know from other studies that it may enhance the inflammatory responses in COVID-19. And so in regions where this polymorphism may be observed, it would certainly be worth thinking about studies that could try and relate this phenomenon of increased inflammatory responses associated with G6PD deficiency to the pathophysiology of COVID-19. Next, please. Well, there are also programmatic challenges that can arise, and one of these could relate to how we diagnose malaria. And here's a recent preprint that's just been accepted, and it describes pre-pandemic sera, pre-COVID-19 pandemic sera obtained in Nigeria, studied on a couple of platforms used for malaria diagnosis. Used for COVID-19 diagnosis. And here it shows that from these malaria endemic areas, there is really quite a high frequency of false positives associated with the presence of anti-Plasmodium IgG levels. So, these lessons and observations cut both ways, and they cut across the different tools that we have, whether it's diagnosing malaria or COVID-19, or indeed, managing patients with one or both of these conditions. Next, please. So just to finish off my talk, I hope we've gained a little bit of time for the panel on this. I just wanted to come back to three key points. One is that when it comes to managing malaria in the time of COVID-19, a lot of the challenges are programmatic. Next point, that it's very important to bear in mind what the science already tells us about the potential for interaction between these two infections. And these can have immediate bearings on interpretation of the tools that we're using. And finally, despite all the challenges of the overwhelming demands put onto healthcare systems, it's remarkable that colleagues around the world are still finding the abilities to answer scientific questions that are of interest to both conditions, malaria and COVID-19. So I thank you all for your attention, and I hand back now to Sachin. Thank you very much.
Thank you, Sanjeev, indeed, for a beautiful presentation highlighting the effect of pandemic on ongoing elimination program, which are getting adversely impacted, especially malaria, TB, and HIV, as you mentioned, and how to sustain the momentum of malaria elimination program. In fact, WHO report quantified the disruptions in malaria control operations. So with this, I would request next speaker, Professor Richard Morde.
Good afternoon.
Professor Richard Morde is the head of the epidemiology department at Mahidol Oxford Tropical Medicine Research Unit, an associate professor at University of Oxford, and visiting scientist at Harvard, teaches Chan School of Public Health, and honorary consultant physician. Over to you, Richard.
Thank you, Sachin, and thank you very much for the invitation to present in this meeting. I'm going to talk in less than 20 minutes about antimalarial resistance, which has been mentioned a little bit by some of the other speakers. So I'm going to focus mostly on the artemisinin antimalarials. Is that the way? So the artemisinins are the mainstay of malaria treatment now for some years. It started with the Chinese herbal medicine for fever around 2,000 years ago. They are the most effective antimalarials with rapid action, broad stage specificity against the parasite. They're easy to administer and they have low toxicity. So in guidelines, they're widely recommended for first-line treatment for Falciparum malaria, so as ACTs, combination therapy, for severe malaria as IV artesunate, and then also for pre-referral rectal artesunate for those who are ill by mouth and severely ill in more remote settings. So these are the first pages of the WHO malaria treatment guidelines in the past few years, and it's been recommended throughout this period. So as we've discussed, there's a plan to eliminate malaria by 2030 in the Asia Pacific, including India. And this plan is heavily dependent on continued efficacy of ACTs. So in 2007, the Gates Foundation made a push towards elimination, and this call has been widely adopted, and lots of effort and funding have gone towards trying to make progress towards these goals. So artemisinin resistance was first described in Western Cambodia in 2009 in Pai Lin. So this is artemisinin monotherapy back in 2009, but since that we've now discovered ACT resistance. So that's resistance to both the artemisinin and the partner drug. So this graph on the left here just shows one of the first studies showing delayed clearance of the parasites by artemisinins in Cambodia. And on the right is a more recent study where we're starting to see this resistance being more widespread, particularly in the Greater Mekong subregion. Since this time, it's spread to all the 5 countries and also to some small areas outside. So we're fortunate now in having a genetic marker of resistance to artemisinins. So this is the Kelch13 marker, so a mutation in this long segment of the genome of P. falciparum, and it's part of the propeller domain. And there's a diagram here on the left to show what that looks like as a protein. So there's a growing list of validated Kelch mutations which are associated with prolonged clearance. There's a list here on the right from WHO from 2017, and there's ongoing work which is expanding this list gradually. So the most predominant and the one that seems to be spreading the most is C580Y. So that's the one that's common in Western Cambodia, and it's since been identified on the Thai-Myanmar border in parts of Laos and Vietnam as well. You can see here just one example of a study showing the growth in the predominance of this particular mutation. So Kelch mutations, looking back at historic samples, there were abnormal Kelch mutations back in 2003, and then the prevalence has gradually increased, particularly this C580Y then taking over in recent years. On the left here, you can see the prolongation of the clearance half-life going along with the increase in prevalence of these mutations. So here's a study which is taken samples from across our region in the GMS, and they found C580Y seems to have spread from Western Cambodia to southern Laos into northern Thailand. And then it's been followed by mutations associated with piperacin resistance, which is the main partner drug at the time this study was done. So DHA piperacin being the first line treatment in this area. So this bar here, the red bar, is all of the C580Y mutations in the samples from these different locations. So this looks like it's a spread of a relatively clonal parasite, which is then taking over from the local populations in these areas. So these borders in the diagram on the right are relatively porous. People cross them frequently and informally, for example, to go to the market. So there's a lot of movement of people across the borders. So this is now being termed ACT failure. So we have confirmed P. falciparum cases where the ACT DHA piperacin does not work sufficiently, doesn't fully clear the parasites, and WHO have started to declare this and keep track of it. So here's a map from back in 2017 when they're showing the number of ACTs which are now failing at more than 10% in the different countries in the GMS. So this ACT failure we're defining here as artemisinin resistance plus resistance to the ACT partner drug. So the concern here is, like this classic map, chloroquine and pyrimethamine resistance also originated in Southeast Asia and then spread into Bangladesh, into India, into Africa. And the worry is that the same is going to happen again with ACT resistance. So WHO are keeping track now. They have the WHO malaria threats map. When there are maps like this which show studies that have been done around the world which try and quantify resistance in different locations. So this particular map showing the prevalence of molecular markers of K13 mutations. And there's also therapeutic efficacy maps and other maps on this website. In parallel, WARN have been developing their molecular surveyor, so the Worldwide Anti-Malaria Resistance Network based in Oxford. This is their website where they keep lots of data from published studies, from trials and observational studies of resistance markers and also treatment failure. So it's a very rich resource. So we've been working on a systematic review to try and put all of the published data together up to date on the CALCH markers in Asia. So it's a systematic review together with WARN. So we're using their database and then also with a systematic literature search, which has expanded the number of studies to 68. So about 10,000 papers have been screened and there's 68 studies included. We've got in contact with many of the authors of these studies to collect additional information, and then we've compiled the data set by year and by subnational unit in all of the different countries. So on the right here is just a summary of the samples collected. So this is the number of blood samples collected per year in these different studies. You can see that it increases quite rapidly up to 2014, and then it's tailed off a little bit more recently. The majority of the samples have been collected in the greater Mekong region, and then a smaller number outside of that area. So we're using this data to produce maps like this, where we've got subnational units in each country by year. So here it's just summarizing with the latest data from each country with the date of collection here, not the date of publication. So you can see that the validated markers associated with artemisinin resistance are commonest in and around Cambodia, southern Laos, northeast Thailand, and a little bit in Myanmar as well. And then very few sites outside of the region who have got any validated markers. There are a few in India, which I'll talk a bit about later. So this is a more detailed map showing the individual markers of artemisinin resistance. So each point shows the predominant marker present in each of these areas. And quite curiously, it's very unusual to find more than one marker in one area. We don't really understand why. And then I've shown the prevalence of each of these markers. So where it's very dark, the prevalence is very high. So here we're looking at the number of the prevalence of the resistance marker over time. So there are some sites where there are samples collected in multiple years. So we've just shown here in black the sites that have samples with at least 3 years of data. And then we've drawn graphs of the prevalence of the validated markers over time for each of these sites. You can see that in many of these locations, the prevalence is increasing. In some, the numbers are quite small and the trend is less clear. So this is South Asia. So validated CALCH markers have only been detected in India in South Asia, and that was in 2012 and 2015. There may well be some new data that wasn't included in this. We're currently updating the search. The validated markers have been found in West Bengal, in Bankura and Kolkata, and then in Arunachal Pradesh in Changlang. And to my knowledge, treatment failure hasn't been reported. So these are just the markers that have been detected. But the concern is, of course, that with Myanmar next door and porous borders again, there's the possibility of spread into India and beyond. You can see on this map that the coloured in districts are those for which there are data, and there's large parts of India where there's no data. And the vast majority of these are just 1 year or one study where there's no serial data over time. So there's a lot of work to do to try and keep track of this and to keep an eye to identify if and when spread occurs, and then also to guide what to do when it does happen. So this is just summarizing all the data together. So there's two different classifications here, the WHO list of validated markers, and then WARN have produced their own updated list, which is a bit longer. You can see the validated marker is increasing in prevalence over time, and then the top bar is just from India and Pakistan, where in 2019 there were no mutations; it was all wild type. But there's this worrying trend of increase in validated markers. So from 2002 to 2018, there's been a steady increase in the number of locations and the proportion of infected people with these validated markers of resistance. The data in the literature are reported very heterogeneously, so it's quite difficult to interpret and to pool them together, this sort of analysis. There's a big need for more consistent data collection over more extended periods in the same locations, and then rapid sharing of that data so it can be acted upon. And there's a need to map the spread and evolution of resistance to inform policy decisions. So this work is currently under review. The main person responsible is my student, Frank Kagoro, and then just to thank the other co-authors and contributors to the work. And if anyone would like to get in touch, my email address is at the bottom. Thank you very much.
Thank you, Richard. Thank you very much. And I would really like to highlight here, despite being ill and having some health issues, Richard is kind enough to give us today's time and today's talk. And thank you very much for highlighting artemisinin resistance, which is a potential threat to malaria elimination program across countries, and the importance of monitoring the resistance markers, especially in Southeast Asia. Thank you very much. With this, this session is over. I know we are running quite late, but without any further delay, I would request all the speakers for a panel discussion. And we have curated the session keeping in mind that we have a variety of renowned malaria scientists across the world, and will definitely be benefited on the question how further future research and overall future research can help overall in facilitating elimination program. And now here, I would request Dr. Manju Rahi to lead the discussion, and I really thankful for her to giving us our important time. Dr. Manju Rahi is a scientist, senior scientist at ICMR headquarters. She is a medical professional with a training in public health, and she is a nodal officer for vector-borne diseases and One Health, with a deep interest in implementation research. Dr. Manju Rahi, over to you. And I also request Dr. Amit Sharma to chip in, please.
Thank you, Sachin. And a big hearty thank you to all the speakers who really have taken time and taken us through the entire journey of malaria, beginning from the program to the basic molecular stuff and to vector control. It's really been heartening, as if I'm studying malaria from end to end. So it was really, it's been the entire afternoon spent on malaria, and one still feels very, very rejuvenated hearing all the success stories and the challenges where we are. So without taking much time, and I've gone through the questions of the audience also, and some of my questions as well. And I think the first and the foremost question will be posed and to Dr. Desh Kumar, who has taken us through the journey of the program and the success. And because we are talking about malaria elimination program, and India is at the forefront for the Northeast, for the Southeast Asia region. And many of the questions have been also on it. But my first question would be, sir, to you, like malaria surveillance has been given a lot of importance in India as well. And as we are nearing the elimination target and in the time frame, and you also highlighted it needs to be a case-based surveillance and involving all the partners and stakeholders in this elimination journey. So what is the plan of NBTCP to take up private sector, just not only taking and notifying, they should notify the data or just, getting their numbers. Is there any other role planned for private sector to chip in the in case management or right diagnosis? Do you plan to involve them more intensely how it is being done in TB program? Are there plans? Second question is about the tools and the which is correlated with the asymptomatic malaria, which you yourself highlighted as a challenge in the coming days when the the obvious malaria will come down, the fever and the symptomatic malaria. Low density infections, asymptomatic infections will require tools other than RDT and microscopy. So in the need of molecular tools, and how do we try to fit these tools into the strategy of the national program? Over to you, sir.
Thanks, Dr. Manju. I don't know the questioner. It would have been much better if the questioner would have been also, indicated or introduced a bit. Anyway, we understand because we are running behind schedule. The first question regarding the role of the private sector, as I said briefly in my PPT, this is of paramount importance. Some efforts have been made towards this. The first step was that we asked all the state governments to make malaria a notifiable disease. So far, 31 of 36 states have made it notifiable. The remaining 5 states are also in the process of making it a notifiable disease. Then the second step was to map all the private sector laboratories, nursing homes, clinics, hospitals, etc., which has been done in some of these states, which is being done in some of these states. And the third step is to acquire or to collect or to ask them to report on regular monthly basis the malaria cases and deaths. The role does not end here actually. The role is much more. We have also spoken to the professional bodies like IMA, Indian Medical Association, IAP, Indian Association of Pediatricians, etc., so as to involve their members in this endeavor, and then to interact with them. I won't say train them because they are already registered medical practitioners, but to sensitize them. And then we also have gone ahead rather than, because if we look at the private sector is basically a formal private sector and a non-formal private sector. Non-formal private sector is equally important, or I would say rather more important. So what we thought is we also mapped the non-formal private sector at the block level. The strategy or the objective was, after mapping them, we can train them, or even we can give them a box of RDT to every non-formal private service provider, and then our local worker can be in touch with them. So whatever cases are diagnosed by him or her, without affecting his livelihood, so if that report can be shared with us. We interacted on this with the IMA, but the IMA was not very supportive on this. The reasons are obvious. But then I countered them because the ASHA is also a kind of non-formal.
True, sir. But
the ASHA is being given the RDT box. They have been given the anti-malarial strips also, and the diagnostic as well as the treatment facilities are being made available at the village. In fact, that is one strategy which has helped us in declining the malaria up to this extent. So all those efforts are there. Then for training, I think it would be worthwhile to share that under the GFTM supported project, we are going to develop an online interactive training platform. If we look at the training load in the country, it is in lakhs of workers, and then the higher ups at the districts, states are needing training. So in that portal, there would be a system so that the private sector can also be involved there. So as of now, it's a comprehensive approach which we are looking at. I hope I have answered the question to some extent or most of the extent.
These are very impressive and very needed steps. And it's really good to hear that all these are being planned and these are the future ready steps, or rather taking along the private sector is important. Sure, sir.
Dr. Rahi, may I request to take some questions from Bart, please? Bart is already late and
Right. Sorry. So these are the next would be to Bart and to Marta combined, because
No, the second, Dr. Manju, sorry for interrupting. I think there was a second question also.
yes, sorry, on the molecular tools and how to adopt in the program. Yeah, Sachin.
Do you want me to answer
Yes,
that
sir.
or skip that?
Yes, sir, we want you to answer briefly.
Okay. Yeah, briefly, of course. See, the asymptomatic malaria is again very important. And we know there are some areas where the asymptomatic malaria prevalence is much more. We have made an attempt in terms of the mass screening and treatment. It's a new strategy which has been added to the actually the earlier strategies. And in some of the high endemic malaria states or specific areas, we have applied this also with some very good results. Examples are Mizoram, number 1, number 2, Tripura, number 3 is Chhattisgarh, and number 4 is Odisha. Now, we definitely require a much more sensitive, much more specific, much more field-oriented tool which can be applied at the field level. But as of now, I would say there is none which can be applied at the field level. We have the tools, but not at the field level. So that kind of input is required for the program, and we are looking forward to the research counterparts on that. I remember one study being conducted in actually Haryana, Mewat area, where some efforts are being made towards that, and we are eagerly awaiting for those results. Till that happens, what we are doing is, to conduct a very fast and small study so as to know the prevalence of asymptomatic malaria in the community, number 1. And number 2, then we identify those areas and then we actually apply the mass screening and treatment strategies at frequent intervals so that the parasitemic load of the community comes down sharply. And then the low endemicity of that area is maintained so that it also becomes part of the less than 1 API endemicity.
True, sir. Yeah, I think all point very well taken, and we are working in that area in that study, hopefully to give you results as quickly as possible within the time frame. With COVID disrupting some work, but hopefully we'll get over that. Thank you, sir.
Thank you so much. Thank you.
Now coming to Bart and to Marta. So you talked about novel vector control tools. Marta talked about repellents, and Bart's talk was more on the larvicides, maybe spread on the water bodies and through. Other than chemical insecticides, looking at other vector control tools and maybe strategies. So vector control is a very important pillar for malaria elimination and eventually eradication. And there is we need to bridge a gap. There is forest malaria, there is outdoor transmission. We do need other vector control tools other than Alliance and IRS. So repellents, ivermectin, ATSB, all these tools are in the pipeline. They are being tested and they are being tried. There are insecticide impregnated strips which are being used somewhere in Thailand. So what is the future of these and how soon can they be ready for adoption by communities or by the countries? Or they always appear very interesting, but how soon can they be ready for the program people like Dr. Desh Kumar to say, okay, now we can take it in the program? So it's a complicated question, but seeing the malaria elimination targets coming very soon, and maybe the existing vector control tools failing us with insecticide resistance, saturations, mosquito changing its behavior, we do need new tools. So how soon or how effective and how successful these can be? I mean, either of you can answer or both of you. Thank you.
Maybe I can start and then Bart can finish. I think that a lot of it relies a little on the WHO. So for a tool to be adopted by a national malaria control program, it has to be pre-qualified by the WHO. And at the moment, the WHO requires evidence on effectiveness from at least 2 trial sites over a period of 2 years. So in regard to all of the different interventions that you listed, we don't have that evidence yet. Ivermectin has just started. There are some trials that are ongoing. I'm involved in one of them. ATSBs, same thing. These trials are ongoing. There are already existing trials on spatial repellents, but as we saw, the evidence was not convincing, and so we can't expect WHO to make a recommendation on that. So yeah, it just depends on how fast investigators and funding can be put together to generate the evidence that WHO needs to pre-qualify these interventions. Over.
Thank you.
I agree with everything that's been said. I mean, that was exactly going to be my answer as well. To give you an example, I submitted a dossier to the VCAG, the Vector Control Advisory Group, that is dealing with all this matter, for Eve tubes now 8 years ago. The first dossier was submitted. The first results from the randomized control trial in Ivory Coast have been published in the Lancet recently, and now there needs to be at least one more trial before there will be pre-qualification. So we're looking at least at another 2 or 3 more years before there's going to be any recommendation from WHO, meaning that you're going to be looking at a time frame of 10 years. I mean, it took for BTI as a larvicide, it took 7 years to go through the process. So these are lengthy processes, and it should be noted that it's not only up to WHO. I mean, ultimately, it's the member states that make decisions as to what they want to use, as to what they want to apply. So if there's a really effective vector control tool that is specific for a given setting or a given country, then by all means, if the country is capable of picking up that tool and disseminating it and using it on a large scale, they should definitely do it, and perhaps in parallel, then submit the dossier to WHO in order to get the pre-qualification. But it is a fact of life that it's a very lengthy and time-consuming process, unfortunately.
Right. Thank you, Bart. Thank you so much for being here. And since Sachin said it's getting very late there, but we really heartily thank you for being here.
Thank you very much. I have to sign off because I have to run for another meeting, but thank you so much for the invitation. Thanks a lot.
Thank you, Bart.
Okay, bye-bye.
Thank you for being here. About the length of time the research takes, and for any, for the extrapolation of the research finding to the real tool, coming to the basic research, I'll pose it to Dr. Das and Dr. Utpal together. I have major interest in public health, but seeing this exciting basic research findings, and I will take support from Dr. Amit in posing these questions. This basic research, and you're talking about the narrative is malaria elimination and eradication. Basic science research is a long journey. Any new drug molecule, new drug target, or vaccine for that matter will take years to come. So how does the basic researcher aims to sustain the interest of a funding organization or a donor in sustaining this kind of research, where an inpatient program, an inpatient public health person would say, where is the applicability of that? You show me a new drug. So how do you sustain the interest, and how soon these may be, alternatives to artemisinin resistant? I mean, artemisinin, and do we have more drugs in pipeline? And Dr. Richard Moore talked about artemisinin resistance mapping. So what are the alternatives, and how far are we from the alternatives, and how long do we need to wait to have new diagnostics, new drugs? Basic science journey is a long one, arduous one. Over to you, to Dr. Ashish and Dr. Utpal, and if Dr. Amit has any additional questions for the basic science.
Okay, thank you for that thoughtful question. Very relevant. This is Utpal here. Am I audible?
You are audible. Thank you.
Okay. Yeah, I think, it's a very complex question, and this is a debate that it can get into a long debate. So, but I will just make one statement as far as my personal understanding of this question, and the answer in my mind is actually very clear. When you work on malaria, there is nothing like a basic science. Everything you do is applicable. It's only the question of time. When is it going to be applicable? So everything that I'm doing, I'm thinking only in terms of application, and it sooner or later, it will make relevance. I'll give you an example of when PCR, we use PCR, RT-PCR. Without that, you can't imagine COVID pandemic times, right? How was the enzyme the enzyme, thermostable enzyme, discovered by a person who identified this protein in what were thermal springs in somewhere in the United States of America. He identified a thermostable protein. So I think that's an excellent example that what has become applicable, as long as basic science is genuine and strong, and something that is, I think that is. But yeah, at personal level, I feel when I work on, and I do work on many infectious diseases, the only question I have in mind is what can I do to create an intervention at one level or the other. Sometimes it's direct, sometimes it's indirect, and sometimes it takes time. But that's my personal view. But there are, of course, people, IISc, for example, is full of, my colleagues are actually very fond of fundamental research, which often doesn't need to be justified by application. There are a large number of scientists in this country who believe that, but I don't believe that. I think that every research has to have application. It's only a matter of time that, and one has to try, look for an application. Thank you, Dan. Thank you for giving me an opportunity to express my views. It is a very nice and pointed question.
Thank you for taking till the application level. Dr. Ashish, you want to supplement that answer?
Yes, I totally agree with what Dr. Upul said. Putting in the caveat that if you look at your tools today, they were mostly the result of basic research some time back. So tomorrow, you don't know. I mean, we all want malaria to be eliminated, eradicated, and everything. Totally for it. We are all in it together. But if you want to have a safeguard, you have to support basic research. Focus basic research with the intention of giving a deliverable at some point in time. It is very difficult to always put a time point on new discoveries or new ideas. Some things will mature faster, some things will mature at a slightly slower pace, depending on how it goes. And you would be the best to appreciate that if you're doing something new, there are risks. Sometimes it may not mature, right? But that's the challenge. I mean, from what matures will come the tools for the future, which is not saying anything against using the tools at present. The tools at the present obviously should be judiciously used, but we all are aware of the loopholes in some of them. So with that, I'll stop. Thank you.
I think totally agree with both of you. Totally agree. We do need continuous R&D to be future ready in case the present tools fail. But my only worry is the time frame where we are, it's a race against time to achieve elimination, and the Lancet Commission talked about eradication. So we need to keep ready. Now, Amit, you want to supplement any questions on that aspect?
Well, I have a huge conflict of interest in this because I will obviously support basic research like my colleagues who've done before me. But I've had the privilege, I should say, or vagaries of life have brought me into a public health institution where public health issues are larger than basic research issues, which is completely reasonable. And I should say that having tasted this side of life, it seems to me that there is a disconnect between hardcore basic research, cutting edge research, and public health issues. They don't necessarily always go hand in hand. There is a bit of asynchrony between the two of them. And this is in context of malaria alone. I can't speak about other diseases. And I think it would be very well taken if in India, some institutions had very strong public health teams running in parallel with very strong basic research teams. The basic scientists need a reality check. They can't just be doing blue skies hoo-ha for 20, 30 years. On the other hand, the public health people need the tools and the deep understanding that basic scientists bring to any issue in order to be able to reveal the trajectories of public health issues. For example, as you know very well, India has missed again the COVID bus, not only missed, but blown up the bus station. And it is being largely targeted because, you know, we missed the double mutant about 6 months ago in September because the genome sequencing was not even being done here or considered. We didn't know the double mutant existed in our society. We probably don't know how many of those mutants still exist to date, and we are trying to make claims about mortality and whether the youth are being affected or not. I think this is all gibberish. So in the absence of hard molecular data, which basic scientists can provide, you can actually mistake and misunderstand and misorient yourself in context of public health disasters like we are going through today. And I think therefore, better synchrony is required. Now, if people like Dr. Utpal Tatu and Dr. Das were part of NIMR, should I say, I think NIMR would be richer. On the other hand, if a few public health people were part of ISC institutions or Blitz Pilani, I think those institutions will be stronger. So we are really talking about cohesion of public health program and research under one wing. That's my thought on this.
Totally agree. We need this convergence, and they don't need to be working in silos, in separate environments and ecosystem. They need to be together. That's a fact. There's a lot of convergence and maybe crosstalk required to remove this disconnect. And I totally agree with the three of you. Now, coming next to Dr. Sanjeev Krishna and to Dr. Avdesh Kumar together, and Dr. Sanjeev Krishna talked about COVID and malaria, the interaction and the not only just the operational disruptions, but the scientific and technical part of it, but coming to the operational part of it, Dr. Sanjeev, can you somehow anticipate what will be the long-term impact of the disruptions caused by COVID in the malaria elimination program and the way it's being rolled out? Even though, I mean, it's been very brave of the program and the entire government, I think, across countries to still continue their work. Maybe the workers are wearing PPE or whatever, but there would be disruptions. And the WHO document which Sachin referred, it showed a range of, you know, from 25 to 75% disruption, depending on how logistics chains would be disrupted, how the RDTs won't be delivered, there would be confounding of diagnosis, there will be fear in the public. These are very real threats of COVID. And looking at India, we are into a second wave. We don't know there would be a third wave, fourth wave, and it's for all the countries, especially the poorer malaria endemic countries. What is the long-term impact of COVID on malaria elimination program? This is my first question. Second question, do you anticipate the infrastructure which has been created by COVID? So, Dr. Ashish, or somebody referred to RT-PCR, or Dr. Utpal maybe, how we have, you know, developed RT-PCR, and now in India at least, I don't know how many thousand laboratories are there now at each district level having RT-PCR machines. Can these structures which have been in an emergency as a knee-jerk reaction created via COVID emergency, but it's a blessing in disguise, we have those infrastructures now, can they be co-opted for malaria later on or parallelly or sometime? Can we have an advantage of COVID infrastructures now? And what are the long-term disruptions? So these for the program benefit as well as, and very pointed question to Dr. Sanjeev. And it was a very thought-provoking presentation. Thank you.
Well, thank you very much for asking the hardest questions to me. You're asking me to predict, you know, what might be happening. I think, of course, the answer has to be very much on a country-by-country basis, because the problem of malaria varies enormously country to country, and is luckily still moving in the downward direction for many countries. And one hopes that momentum that's been generated in the past few years, we know the curves have flattened in the last 3 or 4 years in terms of the numbers of cases, but we hope that the momentum of that downward trend can be maintained despite the short-term threat of COVID-19. We should bear in mind one obvious population effect on the figures on malaria in general, and that is that the population in sub-Saharan Africa in the time that we've been looking at it for malaria elimination has doubled. So even though the numbers of cases may have flattened where most malaria is, really that is a very, very significant increase in the control of malaria and towards its elimination. Now, you know, if people can maintain an integrated approach to malaria elimination, and this comes to your second question really. So if there's some way to integrate thinking in terms of healthcare pathways, infrastructure in terms of hardware and software, actually looking at, and software is very important these days. It's already been mentioned in previous talks, and you know, I like very much the web-based collection of information, because those information portals can be easily adapted to deal with or to adapt back towards malaria when COVID-19 disappears, and it will disappear. You know, these moments should not be lost, and I think that's really what you're also emphasizing, is that there are hidden in these dreadful challenges, there are opportunities also to maintain and sustain longer term impacts on malaria elimination. Now, I'll stop there. Actually, can I just say one thing? Coming back to the length of time between molecular through to applications. 1999, we discovered the glucose transporter of the malaria parasite. Many years, Amit and I have been talking about, you know, crystallizing it and looking at inhibitors and so on. Well, we identified an inhibitor, and it's taken 20 years for another group to crystallize the glucose transporter with the inhibitor, which probably means another 3 or 4 years, 5 years before you can end up starting the drug studies. So sometimes that's how long these things take, from science through to treatment. I'll hand back to you. Thank you.
Thank you. And over to Dr. Ajay Kumar now, adopting the infrastructures not only of laboratory, but as he rightly pointed out, it was in my head when I was framing the questions, like mobile platforms for data capture when we are talking about near real-time capturing of data and surveillance, and we are switching on to some smart surveillance. Now, COVID and Arogya Setu app is in India, and I'm sure many of the web portals and dashboards and mobile apps have been developed for capturing COVID data. Can we adopt some of them for malaria? Is there a future here? So you can unmute yourself.
I'm sorry. Yeah, thanks, Dr. Manju. See, the impact of COVID-19 on malaria would depend not only from country to country, but from state to state also, and from the region to region also. It would also depend on what strategies we are adopting for malaria control or for malaria elimination. Now, coming to, with reference to India, like our main strategy is to make the diagnostic and the treatment services available at the village level. So what we did, or what we advised, and what we continue to actually advocate to the states as well as to the districts, that please maintain the logistic supply at the ASHA level, because ASHA is within the village. Some of the villages were barricaded actually during the COVID time, actually the first wave. So the village heads, they were not allowing others to enter inside the village boundaries. So in that scenario, the best proposition available for us was to strengthen the ASHA, to monitor and to supervise the ASHA, because ASHA was within the village. So if the logistic supply is maintained with ASHA, and these are the ASHA which have already been trained on these things. So that's how we could maintain the surveillance, although there was some decline in surveillance, no doubt. But to me, it looks like that this decline in surveillance was more because of the non-COVID fever was less compared to when there was no COVID.
Okay.
So, but yes, of course, some of the other interventions like IRS, like LLN distributions, etc., were affected or impacted negatively, no doubt. But then there are some best actually practices also which I have shared, like, because it actually varies from state to state. Now, coming to the second issue or the question that whatever infrastructure or facilities which have been made available by the COVID-19 pandemic response, we have now large number of RT-PCR machines like maybe CBNet or TrueNet, etc., etc. And now I think in one district, I think because I know that maybe there are 2, 3, or even 5 machines lying there in some of the districts. So that those machines or that technology can now be utilized for the diagnostic purposes of malaria. What we need is to standardize the SOPs or these tools so that the samples can be collected from the village, and they can be fastest mode if they are transported, and if the report can be made available within maybe 24 hours or so. What we were saying actually earlier in the program, that within a day the report should be made available. I'm talking of those areas specifically which are now low endemic areas.
Right.
Because the RDT or maybe to an extent the microscopy may not be able to diagnose all malaria cases. So yes, that infrastructure can be utilized, definitely. There is one more actually positive coming from the COVID side, because COVID also has some impact on this. Actually, there is, like Asha, there is going to be one more male worker in the village, which would be known as Vishwas.
Okay.
The sooner it happens, because it is under the consideration as of now, so that instead of one Asha in one village, we would have at least actually 2 workers, one male worker and one actually female worker. So I think that can also be utilized for malaria. So I think that would also stand. So these are some of the opportunities which are coming our way, and because of the COVID-19 basically. Thank you so much.
Thank you, sir. I think it's wonderful news, and hearing about a male worker at the village level, in addition to Asha, would be really good. And that will really strengthen the program in general, because Asha, we have heard that she's overburdened and blah, blah. So it will be really good. Good. I mean, thank you for this piece of information.
This concept actually has been going on for last, I think, few years, because there was demand. We have been asking that there should be one more worker who can focus on communicable diseases, including malaria. And then actually this Vishwas is an actually acronym for the Versatile Integrated Social Health Worker for Accredited Services.
Wonderful.
So,
very nice, sir.
So let's hope it gets through quickly, and then we can move ahead, I think, much faster. I mean, so as to stick to the timelines of malaria elimination and maybe other disease eliminations also, like tuberculosis, etc.
TB 2025. Very true, sir.
Yeah.
Before that, even Kala Azar and NF are there. So it's wonderful to hear this piece of news.
Absolutely.
Thank you for sharing this.
But I would say, but this would not be possible without, engagement from the public. As sir said, be a hero, make malaria zero.
Absolutely, and these workers are from the community. They belong to the villages, and that's how the participation of community is, totally on board. Very heartening to hear.
Yeah.
And my last question to Dr. Richard Maud, and then we wrap it up. Last question is, he talked about mapping of artemisinin resistance, and again, it's a broad question. We understand there are 6 combinations of ACT, and many of them are failing, and especially Cambodia, Vietnam, Greater Mekong region, there is a challenge of resistance. What are the alternatives for ACTs, and how soon can we have potent alternatives? I know the 3 drug combinations are being tried. Artemisinin with 2 partner drugs are being tried in some countries to be future ready. But what is your view on having an alternative sooner than later? Because again, it's a race against time for malaria elimination. And once we lose one potent tool like artemisinin, it will be very tough.
Thanks very much for the question. Yeah, I think it's difficult. As you mentioned, there are triple ACTs being trialed, and those trials have a lot of associated studies to try and get them through to approval as quickly as possible, including packaging and manufacture. So the triple ACTs are an option that's coming. They're certainly effective, and they appear to be safe as well. There are some novel compounds under development, but it's a sparse pipeline. I won't go through the list, but there's not that many. and there's still some way to go. Key at the moment is making the best use of what we have. So some countries are switching to different ACTs and then closely monitoring resistance, and then potentially switching again if it starts to increase in prevalence. And that's working to an extent in the Mekong region. As you head towards elimination, it's just test and treat as many people as possible. In most cases, the resistance is a mild phenotype. People do eventually clear their parasites. The severe phenotype's limited to this C580Y in the GMS at the moment. So key is monitoring for resistance and carrying on intensely with current options, I think.
You also mentioned the limited number of sites in India which are capturing this data. I mean, coming back to India, and program is on board with us. So, and I totally get it. The sites are very limited, and which maybe we may be missing the sites. So what, according to you, should be the plan, and how do we supplement that information from India? Do we just increase the number of sites, or we have more methods of sample collection like DBS across the country? What will be the best method to supplement those sites?
Yeah, it's a good question. It's typical in many countries that most of the data, if not all of it, comes from research projects, which are short-lived in particular single or a small number of sites. So the vast majority of what are presented is that sort of data. Some other countries, much smaller than India, have made it part of surveillance. So there's a big project we're supporting at the moment in Laos, Vietnam, Thailand, Cambodia, where they're routinely collecting blood spots from every patient. And then it's just a matter of having the right capacity to be able to process those blood spots. So we're at the minute sequencing parasites in the Sanger in the UK, but ideally want to have local capacity where that can be done quickly and efficiently and high quality. But it's becoming very cheap to sequence these parasites, so it is feasible.
Okay, so you're suggesting part of the routine surveillance, and same thing also goes for entomological surveillance and insecticide resistant surveillance. They need to be part of the routine surveillance, then you capture more of it.
Yes.
Great. Thanks for that answer. And that's right, spot on. And that's how eventually it will become, but maybe right now we are just on the therapeutic efficacy studies, which are taken up every 2 years at very limited sites. But I think eventually we would need to go into that mode of routine surveillance on larger geographical areas. I take that point. And I think we have covered an entire, domains, very different domains of malaria. And that is reminding me of the One Health approach, which we have for zoonotic infections and all these re-emerging infections, how to take all partners on board and talk at the same time. So I think this was an excellent example, today's symposium, where from basic to operational to the program, we had the entire spectrum covered. Over to you, Sachin. And thank you for all the patience you had, during this panel discussion, and it was good. I just picked up a few questions from the, from the audience, and some of them we thought about them. And but good to hear that COVID has a silver lining where we can pick up few lessons and adopt on malaria. So ending with that positive note, over to you, Sachin, for the wrap up. And thank you once again from my side.
Okay, yeah. Thank you everyone for your time and patience. My special thanks to our distinguished speakers. They have taken out their time from their busy schedule and accepted our request. I will extend my thanks to MERA-India team and NMR team. And also a big thank you to Ms. Rosalind and Dr. Prashant Mathur for the Zoom webinar facility. Thanks to Dr. Manju Rahi and Dr. Amit Sharma for their wholehearted support and guidance. I really appreciate patience and the presence of all the participants. And I think we'll come up with such more meetings in future. Thank you very much.
Thank you, everybody. Bye-bye.